Hello. Hello. हॅलो.
सर तुमचा
येतोय. आवाज ऐकू येतोय सर?
हो आवाज येतोय सर. आवाज
आणि स्क्रीन पण दिसते?
हो हो.
Shall we start now?
या सर.
So good afternoon to all of you. Myself, Dr. Pritam Shinde. I am working as a assistant professor of agriculture botany in College of Agriculture, Pune. Previously, I was working at College of Agriculture, Dhule at Agriculture Research Station. And I was also a student of this College of Agriculture during my UG BSc Agri. So the topic allotted to me from your coordinator is regarding to cell organization and different cell divisions. So what are the different mitosis and meiosis, as well as we will discuss something about the linkage and how to estimate and how we identify the linkage, what are the different types of linkages. So basically the cell is the basic component of any living organism. So the cell may be unicellular or it may be multicellular. The organism will have only one cell or they may have different cells. The structure of cell is very dynamic in nature. It is very diverse, but across the organisms or across the different individuals or across the different species, the cell is having some common characteristics among them. And that's why we can, the cell theory which was proposed, the cell theory states that these all organisms, they are composed of one or more cells, and the cells are the basic unit of structure as well as they are the basic unit of function in any organisms, and all cells, they come only from the other cells. So this is very important. The whatever cells are there, they are produced by the different cells which are present already into that particular individuals. So there are two categories of cells: prokaryotic cells and eukaryotic cells. So what is prokaryote? The lower organisms like fungi, bacteria, we call them as the prokaryotes, and whatever the animals, plants, animals, and the higher order of fungus, they are included into the eukaryotes. So there are two basic differences between the prokaryotic cells and eukaryotic cells. The prokaryotic cells, they don't have any nucleolus or other membrane bound organelles. So whatever organelles present in a eukaryotic cells like nucleus, endoplasmic reticulum, or anything, any organism. So in eukaryotes, all these organelles they are present and they have a very big membrane to surround those organelles. Also, there is a one major difference between the DNA structure of these prokaryote and eukaryote cells. The DNA structure of the prokaryote cell, it is a circular type of DNA, while in case of eukaryote cell, you have a linear chromosomes. So these are the basic two structures. Hello. Hello, any difficulty?
So please continue.
Okay, so these are the two basic differences between the prokaryote cells and eukaryote cells. So maybe in a certain objective type of questions, the question may be asked on what is the nature of DNA in case of prokaryote cells. So you have to choose the right option, that is circular type of DNA. Then we are we will just concentrate on eukaryotic cells. So all cells in the eukaryote cells, they are surrounded by a membrane called as a plasma membrane, and these eukaryote cells have membrane bound intracellular organelles. So there are different organelles present in a eukaryote cells. The most prominent organelle in a eukaryote cell is called as the nucleus, and this is controlling all the working of the cell. So it is the main part of the any living cell, the nucleus, because nucleus is the major portion which controls the other organelles in the cell, and that's why the day-to-day activity of any living organism, it is dependent on the nucleus. So animal cells and plant cells have different organelles. So organelles, they have the specific functions allotted to each organelle. The organelles of animal and plant cells, they are very similar to each other, but there are certain exceptions to their similarity. The major exception or major difference between the plant and animal cell is that the centrioles are only present in case of animal cells, while the chloroplast, the plastids, what we call as the chloroplast, are only present in the plant cell. So this is the major difference between the animal cell and plant cell. So you should be aware of these two things. One can ask you what is the basic difference between the animal and plant cell. Although both are eukaryotic in nature, but the presence of centriole, it is only in case of animal cells, while the presence of chloroplast is there only in case of plant cells. So if you see the structure of plant cell, so you will see different organelles present in the plant cell. The total plant cell, it is surrounded by the cell wall, and inside the cell wall you will get another membrane, that is the middle lamella. So cell wall is consisting of three different layers. So these are different mitochondria is present, then Golgi apparatus, then there is a main part, that is nucleus. Then again nucleus can be divided into these nucleolus. Then nuclear envelope is another part. Then chromatins. So these all constitutes the nucleus. So again there is a one bigger central vacuole is there. In plant cell, these chloroplasts are present and ribosomes are there. And all these organelles along with these mitochondria, they are embedded into a matrix. So this is smooth endoplasmic reticulum and rough endoplasmic reticulum. So one by one we will see what is the structure of each of the organelle in the plant cell and what is the function allotted to that particular organelle. So you must have till now we have discussed what is the importance of cell, what are the different categories of cell, prokaryote and eukaryote, and how the plant and animal cell, they are different from each other. So for the benefit, for the sake of latecomers, I am just recapping the whatever discussion still we have discussed about these things. So first part of any cell, it is a cell wall. So it is the outer covering of the cell that protects the plant cell and give its shape. So this is the cell wall. If you study the cell wall in a higher plant cell, it is again differentiated into three different parts. So first is the middle lamella. Then other two parts are primary cell wall and secondary cell wall. The middle lamina, it is the common layer between the two adjacent cells. So if this is one plant cell, so it is having another plant cell alongside with the plant cell. So when these two plant cells are present, these are interconnected with each other with the this middle lamella. So this is the portion of cell wall. So it is the common layer between the adjacent cells. What is the composition of the middle lamella? It is composed of different calcium and magnesium ions. So these calcium and magnesium ions, they are essential for joining together the two adjacent cells. So when there is a disintegration of these calcium and magnesium cells due to the pectin activity or due to the pectic acid, so the different enzymes which dissolve the pectin, there will be a dissociation of the different cells in the cell wall. So when we see that when the fruit is mature, when the fruit is immature in size or it is in immature in stage, then these calcium and magnesium ions, they are present in the cell wall in the middle lamella. So you will see that the coat of the any fruit will be very hard. But once it is in ripening stage or when it is harvested from the plant itself, then the different enzymes, they will have the certain reaction on the calcium and magnesium ions. So that's why there is a disintegration of calcium and magnesium ions in the present in the middle lamina, and that's why the wall of the any fruit will become soft in nature. So this is the basic scientific reason behind the ripening or softening of the skin of the any fruit. So the main reason is the presence of calcium and magnesium ions which are present in the middle lamina. Then there is primary cell wall. So this is the layer which lies between the outer middle lamina and inner secondary cell wall. It is consisting of different cellulose and hemicellulose materials and along with the pectin substances. So actually it is nothing but the network of different hemicellulose microfibrils which are present in a transfer form at the outer surface, while they are present longitudinally on the inner surface of the primary cell wall. And between the two layers of microfibrils, there is one layer of pectin is present. So this is the structure of the primary cell wall, which is just lying between the middle lamella and secondary cell wall. And the secondary cell wall is the innermost layer. So it contains cellulose microfibrils, and the lignin and submarine are also found in the secondary cell wall. So this is the structure of cell wall. It contains three layers: middle lamella, primary cell wall, and secondary cell wall. Middle lamella contains the calcium and magnesium ions which are useful for connecting the two adjacent plant cells, while the primary cell wall mostly it will consist cellulose and hemicellulose material. And it is what is the function of cell wall? It determines the shape of the cell. It gives the protection and support to the cell, and also it will withstand the different turgor pressure created inside the cell. So inside the cell of any plant cell or any animal cell, different biochemical reactions will be going on. So different biosynthesis of certain proteins or fats, disintegration of different carbohydrates items will be there inside the cell, and most often the turgor pressure will be created inside the cell. So the main function of the cell wall, it will withstand the turgor pressure inside of the cell. So even if the pressure is created from inside of the cell wall will not allow to cell to tear into pieces. So this is the main function of the cell wall. So any doubts regarding to this structure of cell wall and what are the functions of cell wall? Then there is another membrane called plasma membrane inside the cell wall. So this is the thin and very semi-permeable membrane that surrounds the cytoplasm of a cell. So the whatever organelles present in the cells, they are embedded into the cytoplasm of a cell. And this plasma membrane is the thin or very semi-permeable membrane that surrounds the cytoplasm. So this plasma membrane, which it will consist mainly phospholipids such as lecithin, and it is also consisting of three layers. The outer and inner layer is a protein layer, while the middle layer is of a lipid layer. So all these three layers are called as the unit membrane. So all different organelles present inside any cell, they are having such type of plasma membrane. So one plasma membrane is already present inside the cell wall. So it is actually the outer boundary of the cytoplasm. So this is one of the major plasma membrane present in a cell as a whole. But the different cell organelles like nucleus or endoplasmic reticulum or Golgi bodies or mitochondria, they also have their own plasma membrane which is enclosing the cytoplasm content of each of the organelles. So this three-layer structure plasma membrane is present in every organelle, and these three layers, they are consisting of outer and inner layer is of protein content, and the middle layer is of a lipid content. The function of the plasma lemma or plasma membrane is that it regulates the passage from inside and outside of the cell. So various chemicals, have to be imported into the cell, or some of the chemicals or biosynthetic product have to be exported from the cell outside the cell. So this plasma membrane is the semi-permeable membrane. So it will regulate the passage of different biosynthetic products inside and outside of the cell. Also, it will check the entry of the different toxic elements. It will only allow one passage to the minerals. So there is only one passage of the mineral inside the cell, but it will restrict the exit of the cell. So this is the content and function of the plasma membrane. Then next important part of any plant cell is the nucleus. So it contains almost all the genetic material is enclosed in the genetic material. So it is what we call as the nuclear genome. So there is a outer covering of each nucleus which is we call as the nuclear envelope. So it is a double membrane structure. So the space between the outer membrane and inner membrane of the nucleus is called as the perinuclear space, and there are different nuclear pores are there. So on the outside of the nuclear envelope, at certain spaces there are certain nuclear pores are there. So this function of these nuclear pores is that they allow the passage of macromolecules or ribosomal subunits inside and outside of the nucleus. So although the nucleus has its own nuclear envelope, but that nuclear envelope has certain spores or some certain pores in between, and they will permit the exchange of material from the nucleus to the outside cytoplasm. So the space between the outer and inner layer of the nuclear envelope is called as the perinuclear space. So one may ask you such type of objective question. The space between the inner and outer layer of the nuclear envelope is called as dash dash. So there are maybe certain alternatives. So you have to choose the right objective answer, that is perinuclear space. And what is the function of the nuclear envelope? It will protect the chromosome from the cytoplasmic effect. So chromosome, which is the controlling unit or which contains the all genetic material inside the nucleus, that is protected by this nuclear envelope from the cytoplasmic effect. Then the nucleolus is the very important part of the nucleus. So it is a spherical body found in the nucleus, and it is attached to the specific region of the particular chromosome. So it consists of three units: granules, fibrils, and matrix. The function of the nucleus is that it will be helpful for the formation of the ribosomes, and the whatever synthesis of RNA is there, that is controlled by this nucleus. What is the process by which the RNA is synthesized? Can anybody tell me? If DNA gives rise to RNA, that particular process is having a specific term, whether it is transcription or translation. So the synthesis of RNA, what we call as a transcription, that is going on inside the nucleolus. Then third important part of the nucleus is chromatin. So it is a tangle mass of nuclear chromosome. So all the nuclear chromosome, it is just like a tangle mass of any thread. So it is the basic unit of chromosome, and it is very important part for inheritance of the character from one generation to another generation, because as you know that whatever genes or whatever the gene sequences, they are present on the chromosome, and chromatin is the basic unit of the chromosome. So this is all about the nucleus. Then the next important part of the plant cell is the plastids. So plastids are the self-replicating organelles found in plant cell. There are three types of plastids are there: leucoplast, chromoplast, and chloroplast. While the leucoplast they are colorless in nature, and they are important as far as the storage of fat and protein is concerned. While the chromoplast are not colorless, they are having color, but it is something different than the green in color. So chromoplast, they are green. Chloroplast they are green in color, and they are very much important for the carrying out the associated with the photosynthetic activity. So the chloroplast is one of the plastid. The other two forms of plastids are leucoplast and chromoplast. Leucoplast, they are colorless in nature. Chromoplast are having some color, but they are different than the green color. These three types of plastids, they are developed through the proplastids. So the basic source of plastid formation is proplastid, and they are interchangeable. So the leucoplast can be converted into the chromoplast, or it can be chloroplast can be converted into the chromoplast. So they are interchangeable in nature. So these plastids are interchangeable. So most important plastid which we in the plant cell, it is a chloroplast. So this is the structure of the chloroplast, which is having the outer and inner membrane and different grana and thylakoids, that is stroma and grana are different parts of this chloroplast. So this is chloroplast consisting of the three main components. First is the envelope. The entire chloroplast is bounded by an envelope which is made up of a double unit membrane. The stroma is the matrix or stroma. It is the part of the chloroplast which is a gel fluid-like basis. So it is a completely a gel-like structure, and it is surrounded by thylakoids, that is grana. It contains different proteins and ribosomes, and this stroma is the unit for CO2 fixation. So this is related to the dark reaction in the photosynthesis. The chloroplast stroma, it is related to dark reaction in photosynthesis, while the thylakoids, they are consisting of the closed vesicles arranged in a membrane network, and they are stacked like a pile of coins. So they will form this grana, and this is important for light reaction in a photosynthetic mechanisms. So the grana are the different granas present, they are interconnected. So these are the different grana, and this is the stroma, which is a fluid-like structure. So in stroma, there is a presence of different enzymes which are required for the dark reaction. So the dark reaction of photosynthetic, that will occur in the stroma portion of the chloroplast, while the light reaction, it is occurring in the grana portion. And these different grana units, they are interconnected with these grana lamellae to each other, stroma lamellae. So these stroma lamellae are present, which interconnect these different stroma and grana unit, and these all group of units which contains both stroma and grana lamellae, they are known as the contasomes, and they are associated with electron transfer and photophosphorylation in case of different photosynthetic pathways. So this is all about the structure of the function of structure of chloroplast, the function of the chloroplast, the light reaction, different enzymes required in the light reactions, they are present in the grana, while the dark reaction, that is Calvin reaction, also present. The different enzymes, they are present in the chloroplast. So it is important for photosynthetic. Then another structure is the mitochondria. So the mitochondria, it is having different membranes. First is the outer membrane, then inside there is an inner membrane, and this inner membrane has different inside folds within that inside membrane. So this is called as the cristae. So the series of inside folds in the inner membrane is known as the cristae, and the space between the different cristae into the inner chamber is called as the matrix. So this is the whatever space inside the two cristae that is filled up with the fluid-like structure, that is what we call as the matrix, and the different enzymes related to the Krebs cycle, they are present in the matrix of the mitochondria. What mitochondria is also called as the powerhouse of the cell. So it is important function. It is having the synthesis of ATP molecules is done in mitochondria. So different Krebs cycle or ETC system or phosphorylating systems are there. So this is that's why it is called as the powerhouse of the cells. Then the mitochondria is important for extramitochondrial inheritance. So different DNA content is some DNA, some extra chromosomal genes are also present in mitochondria. So nowadays it is gaining a lot of importance. The extra chromosomal DNA content present in the mitochondria. Also, it is important for synthesis of different proteins and mitochondrial DNA as well as RNA. So these are the functions of the mitochondria. Then there is endoplasmic reticulum. So endoplasmic reticulum, it is just attached to the nuclear envelope. So this is the nuclear pores and this is nuclear envelope, and through this nuclear envelope it is attached one part, that is what we call as the endoplasmic reticulum. So endoplasmic reticulum, it is connected to the nuclear envelope. There are two forms of endoplasmic reticulum, rough endoplasmic reticulum as well as the smooth endoplasmic reticulum. So the smooth endoplasmic, it is related to the synthesis of lipids and oils, while the rough endoplasmic reticulum, it is useful for protein synthesis. So there are different structure present in the endoplasmic reticulum: cisternae, tubules, and vesicles. These are the basic structural units present in the endoplasmic reticulum. Then there is another part, that is what we call the Golgi network. So whatever proteins or lipids they are synthesized in the endoplasmic reticulums, they will be packed inside the transport vehicles through this Golgi network. So it is just whatever protein or fat molecules they are synthesized in the endoplasmic reticulum, that will be packed into these transport vehicles present in the Golgi network, and they will be transported from one portion of the cell to the another portion of the cell through this Golgi network. So these are the transport vehicles, vesicles. So Golgi network is useful as a transport vesicle in a plant cell, and the larger part of the any cell, it is having vacuole. So it contains a vacuolar membrane, that is tonoplast, and it may contain different water, organic acids or inorganic acids, sugars, enzymes, and secondary metabolites which are required for cell enlargement. And the turgor pressure which is generated by these vacuoles, it will give the structural rigidity to the plant cells, so that the plant will maintain its shape and size. So these are the functions of the vacuoles present in the plant cell. So almost 80 to 90% of the plant cell is in of vacuole. So these are the different parts of the plant cell. So any difficulty regarding to the parts of plant cell? यात काय कोणाला अडचण आहे? So if there is no difficulty, then we will shift to our next topic, that is cell division. So there are different two types of cell division. You might have studied these divisions from your 12th standard biology group. So mitosis and meiosis. So they are, every cell is destined to divide, progress through a series of stages known as the cell cycle. So the cell cycle is the most important part in any cell life cycle, because cell cycle is the process through which there is a multiplication of cell. So if the individual has to survive, he has to have the ability of division of cells. So it will be done through mitosis or meiosis. But But before mitosis or meiosis, there are certain stages occurring in a cell cycle. The first stage of the cell cycle is called as the G1, that is gap 1 stage. So at this stage, it is necessary where a cell has to divide, and if a cell has to give birth to a new daughter cell, then there should be a multiplication of the two parts of the cell. First part is the cytoplasm, and the second part is the genetic material. So what is the genetic material of the cell? That is DNA. So if the plant cell or any cell, by that matter, if any cell has to divide, first it has to increase the quantity of these two, cytoplasm as well as the genetic material. So gap 1 is the first stage of cell cycle where there is an increase in the cytoplasm or protein content. So although it is a resting stage, we call this G1 as a resting stage, but that during this resting stage, there is a increase in cell size, in cell cytoplasm content, in cell protein content. Then after this G1 or gap 1 stage, the cell will enter into the synthesis stage. So in the synthesis S stage, what will happen? As there is an increase in cytoplasm, in the interphase, the DNA will be replicated. So you will have the duplication of DNA in the interphase. So after interphase, there is again one G2 phase that is very short in time, but it is also a resting stage. So during this stage, the whatever cell is there, it will prepare for the division. So it will have the certain mechanisms occurring in the cells, the centrioles or the MTO microtubule organized centers in case of plant cells, they will replicate. So the cell will prepare itself for the meiosis or mitosis during the gap 2 phase. So the interphase is the stage where there is actual DNA replication is happening, and mitosis and meiosis are the two different processes where there is a actual formation of daughter cell from the parent cell. So mitosis, this is the cell division which occurs during the G1 phase. Then the cell advances to the S phase. So here the two copies of the replicated chromosomes, they are termed as the chromatids, and these chromatids, they are joined at the centromere to form a sister chromatid in case of S phase. Then after going through this G1, S and G2, these all are collectively known as the interphase. And then there are different phases of mitosis. First is the prophase, then metaphase. Metaphase maybe can be divided into pro-metaphase or early metaphase or metaphase, then anaphase and telophase. So these are the phases of mitosis. So in the first interphase, the chromosomes, they will start to decondense. So they are in decondensed stage. But at the end of this interphase, that is at the end of G1, S and G2 phase, the chromosome have already been replicated, and the centrosome will be divided into two parts. So the nuclear envelope will be fragile in nature, so it will be start to disintegrate the outer membrane of the nuclear envelope, and the chromosomes, they are decondensed in the decondensed form, so they are in loose form. So at the prophase, the microtubules forming mitotic spindles. So these first is the microtubules, they are form the mitotic spindles. So they will try to separate on the different poles of the cell. The nuclear envelope will dissociate into the smaller vesicles. The centrosome, they will separate to the opposite poles, and the mitotic spindle apparatus is formed. So this mitotic spindle apparatus will be formed, and these chromosomes can be clearly seen, and you can see these the two sister chromatids. So they are attached at the point. So these pairs of sister in the next metaphase, these pairs of sister chromatids, they will align themselves along a plane called as the metaphase plates. So due to the microtubule kinetochore microtubules, they are attaching at the center of the each chromosomes. The chromosomes, they will be placed at the one plane that is called as the metaphase plate, and each pair of the chromatids is attached to the both poles by these kinetochore microtubules. So in the metaphase, the chromosomes, they will line along a plane called as the metaphase plate. So this is the second stage in the mitosis that will complete your metaphase. So there is a spindle apparatus is in complete operation. So the next phase is the anaphase. So during anaphase, there will be a separation of the different sister chromatids. So these sister chromatids, they will be pulled towards the opposite poles through the spindle fiber network. So these spindle fibers network at opposite poles, they will try to separate the sister chromatids along with this microtubule kinetochore microtubule. So this at anaphase, there is a complete separation of the sister chromatids from each other. So one set of the sister chromatid that will go to the one pole, and another set of the sister chromatid that will goes to the another pole. Then at the last phase is the telophase. So these chromosome, they will reach to the respective poles and they will decondense. So there will be a decondensation. Again, the nuclear membrane will be formed to form the two separate nuclei, and in most cases the mitosis is just followed by the cytokinesis. So cytokinesis is nothing but the division of cytoplasm. So in case of animals, it will be done through the formation of cleavage furrow, or in case of plant cell, there is a formation of cell plate. So these two daughter, from the one cell you will get these two daughter cells through the process of prophase, metaphase and anaphase, and at the last you will get the telophase, where the chromosomes will be separated at each pole, and there is a cytokinins which follows this telophase, and there is you get the two daughter cells. So these ultimately produce the two daughter cells, but in case of mitosis, the same number of chromosomes present in the mother cell and daughter cell. So they are genetically, they are identical to each other. So there is no crossing over or there is no recombination occurring in case of mitosis. Only from one cell you are getting two cells. There will be no change in the genetic material. The number of chromosomes will remain same for the parent cell as well as the daughter cell. So that's why this mitosis ensures the genetic consistency from one cell to the next cell, and there is a development of different multicellular organs occurring through the process of mitosis and cytokinesis. So this mitosis is happening in case of different cell organelles, like in case of man, our different organs, they will have the different shape due to the mitosis. So it is a mitosis occurring in in a cells which are not involved in a sexual reproduction. So that's why there is no recombination or no crossing over occurring in case of mitosis. The chromosome number will be stable after the mitosis also. So these are the four stages of mitosis. So one may ask you, in which stage there is a formation of metaphase, at which stage the chromosomes, they will lie on the equatorial plate, or what is the function of these microtubules. So kinetochore microtubule, what is the function, or how the cytokinins is occurring in a plant cell. So those type of question can be asked in your objective type of question paper. So this completes your mitosis. Then there is meiosis. So in somatic cells, these cells, they divide through mitotically and they make up the vast majority of organism tissues. But there is some different mechanism going on for the division of the germ cells. So these are germ cells, they are specialized in the production of gametes, and they are undergoing meiosis to produce the haploid gametes. So gametes, they unite with the gamete from the opposite sex to produce the diploid offspring. So in case of meiosis, from the diploid parent cell, you will get, at the end of meiosis, you will get the haploid gametes. So it is present in case of germ cells. So gametes are typically haploid in nature. They are 1 in number, while the diploid cells, you can see the 2n. So a diploid human cell contains 46 chromosomes, while the gamete will contains only 23 chromosomes, because it is diploid, and 23 chromosomes will be haploid in nature. So in meiosis, haploid cells are produced from the diploid cells. So there is a chromosome must be present. The chromosome number is reduced to the half of the original number. That's why meiosis is also called as the reductional division. So one may ask you, the meiosis can also be called as the dash division. So you should answer it is a reduction division. So in case of chromosome meiosis, the chromosome there that will replicate only once. So it will replicate only once, but there is a division of nucleus twice. So from 2n in meiosis 1, there will be no duplication of the chromosome will duplicate in case of this first 2n. Then there is no further duplication of the DNA. Only your chromosome or the nuclei will divide twice. So from one cell, you will get 4 haploid cells at the end of the meiosis second cycle. So the meiosis is completed into 2 stages, meiosis 1 and meiosis 2. So what are the stages of meiosis? In prophase 1, there is a pairing of homologous chromosome is occurring. Then at metaphase 1, then anaphase 1 and telophase 1. Then there is a intermediate stage, that is interkinesis. The cell will divide, but there is no duplication of chromosomes. So the DNA replication will not be there. Only the division of cell is there. Then the cell will enter into prophase 2, metaphase 2, anaphase 2 and telophase. And at the last, you will get the cytokinesis. So these are the different stages of meiosis. So in case of stage 1, that is prophase 1, it will consist the multiple substages: liptotin, zygotine, pachytine, diplotin and dikinesis. So these are the 5 different substages of prophase 1. So during this liptotin stage, there is a thickening of thin chromosomes occur. So whatever chromosomes are there, they will become very thick in nature during this liptotin stage of prophase 1. At the zygotine stage, these homologous chromosomes, they begin attaching to each other by the one common complex, that is symptosomal complex, which already exists or which exactly aligns the chromosome to the homologous chromosomes. They will be aligned to each other point to point during the zygotine stage. Then during pachytine stage, there is a completion of the symptosomal complex to form a bivalent chromosome structure, and actually crossing over occurring during this pachytine stage. So the crossing over is actually occurring in a pachytine stage of prophase 1 in case of meiosis. Then afterward, the diplotin stage, there will be the disintegration of the symptosomal complex and the separation of homologous chromosomes. And these separated homologous chromosomes, they will again condense in the size, and there is a thickening of the chromatids. So these are the 5 multiple stages of prophase 1, where in case this is the liptotin stage. So the chromosomes, they will become thickened and they will be visible under the microscope, and the centrosomes, they begin to move towards the opposite poles. So this is the first prophase 1, that is liptotin. Then at the zygotine, the homologous chromosomes, they will have the synapses through the symptosomal complex. The naturally the homologous chromosomes which are present in the nucleolus in the zygoting stage, they will come towards each other and they will form a synthesis. So they will form a symphotemular complex. And due to that, you will see that the tetrad of, so each chromosome is having two chromatids. So the two chromosomes have four chromatids in common. So you will see a tetrad like bivalent in case of this zygotine and pachytene stage. So at the pachytene stage, the synapses will be completed, and at certain point attachment of these homologous chromosomes, there will be an exchange of genetic material from the non-sister chromatids of homologous chromosomes. So these are called as these two chromatids of one chromosomes, they are sister chromatids, and these are the two sister chromatids of another chromosomes. But due to the crossing over, there is a exchange of material between these non-sister chromatids. So that's why you will get different genetic recombinations after the meiosis. So why there is in mitosis you will not get any genetic recombinations, because there will be no crossing over or there will be no genetic exchange between the two chromatids of a homologous chromosome. But in case of pachytene stage of the prophase 1, there will be a genetic exchange between the two non-sister chromatids of homologous pairs that will occur during the pachytene stage. Then there comes the diplotin stage. So at this diplotin stage, already the crossing over has already been occurred. There will be a exchange of the genetic material, and the crossover point will be, there will be a tetrad of four chromatids can be seen, and the crossover points, they appear as the chasmata which hold the non-sister chromatid together. So at this point, these are called as the chasmata. These are actually the point of crossovers. So these will be appear as a point of crossover points, and they will hold the non-sister chromatids together. So these non-sister chromatids, they will be held together at this point of chasmata. So the spindle apparatus will again form, and the spindles, they will be attached to the centromere of each of the homologous chromosomes, and they will try to pull the homologous chromosomes at every pole. So during this dikinesis stage, the chromatids, they will be thickened and shortened, so their size will be reduced and they will be very short in size, and that's why they will be more thick in nature. And at the end of prophase 1, the nuclear membrane, it will be completely broken down and the spindle begins to form. So the spindle apparatus will be formed at the dikinesis stage of the prophase 1. So this completes your prophase 1. So at the crossing over, it is an event where the homologous chromosomes, they exchange part between between two, and that's why they will create recombinations of gene and genetic alleles. So if the maternal chromosomes gene content is ABCD and the paternal chromosome genes are having ABCD before crossover, then after crossing over, there may be a exchange of material from the non-sister chromatids. So the maternal chromosomes genes may change to different genetic constitution. So that's why you can get new genetic combinations through the meiotic division. So this is happening in diplotin and dikinesis. Then after prophase 1, there is a metaphase 1. So as we have discussed in mitosis, same way the metaphase 1, the tetrads, they will line up against the metaphase plate. Each chromosome of a homologous pair, that is attached to the primer from the opposite pole, and these sister chromatids attached to the fibers from the same pole. And at the anaphase, the centromere does not divide. The chasmata migrate up to the chromatid ends, and the homologous chromosomes, they will move to the opposite poles. But these homologous chromosomes, they are containing some recombinant type through the crossing over. So these are not the sister chromatids, but some part of the sister chromatid, it is replaced by some of the portion from the non-sister chromatid at the anaphase 1. So this is the completes your meiotic division first. Then at the end of anaphase 1, the homologous chromosomes, they will move to the opposite pole of the cell, and there will be a telophase 1. So after telophase and cytokinesis, the chromosomes, they will gather at the pole of the cells and the cytoplasm will divide. So here, whatever the chromosome number inside the first parental cell, that is divided into half of the number. So here the number of chromosomes will be divided into two parts. So if in case of human being, so 23 chromosomes, they will have the 23 pairs, one daughter cell, and the other cell will have the 23 pairs of the chromosome. And then starts your mitosis second stage. So in mitosis second stage, prophase, metaphase, anaphase, and telophase 1, telophase 2, and cytokinase, they will follow the simple procedure of mitosis. So a new spindle formation across the chromosomes. The metaphase 2 chromosomes, they will line up at the equator and they will be divided, and there is a development of two daughter cells from one parent cell. So you will get 4 haploid cells in case of meiosis from the one parent cell. So this is the reductional division. So here the chromosome, we have discussed that here there is only once the chromosome replicates only one time, and the nuclei will divide into twice. So there is a chromosome duplication. So from one chromosome you will get duplicate chromosomes, and these duplicate chromosomes, they will enter into the two cells, and there is again a division of nuclei. So from two chromosomes you will get from two sets you will get a single set of chromosomes. So this is what we call as the reductional division, and it is occurring through prophase 1. The prophase 1 is containing 5 stages: leptotene, zygotene, pachytene, diplotin, and dikinesis. Then metaphase 1, it is followed by the prophase 1 is followed by the different stages: metaphase 1, anaphase 1, and telophase 1. And after telophase 1, the cell will enter into the prophase 2, metaphase 2, anaphase 2, and telophase 2, and then cytokinase. So this is all about the mitosis and meiosis. So hope you might have understand these things. Although it is in a short span of time we have discussed all these things, but with the help of figures, I have tried my level best to describe each and every stage in case of meiosis and mitosis, as well as the cell structure. Then the last point which is given to me is the summary. What is the difference between the mitosis and meiosis? So there is one round of DNA synthesis in the mitosis. Also in case of meiosis, there is one round of DNA synthesis. But in case of mitosis, there is only one cell division occurring. But in case of meiosis, two successive cell divisions are occurring. It will produce two somatic cells, while it will produce from one cell you will get 4 germ cells. There is no independent assortment. So there is an independent assortment. It will produce the diploid cell, it will produce the haploid cell. No crossing over is occurring in case of mitosis. The crossing over is occurring in prophase 1 at the one of the stage, and it is useful for growth and cell replacement and for asexual reproduction, while meiosis is important for sexual reproduction. Then what is linkage? The topic allotted here was also linkage, or the tendency of genes to remain together in their original combinations during their inheritance is called as the linkage, and this phenomena was first reported by Batson and Punnett in 1906. And there is chromosomal theory put forth by several, stated that the many genes, they are located in each chromosome in a linear fashion. So what are the genes located in a single chromosomes, they will move to the same pole during the cell division, and as a consequence, the genes will follow the independent assortment and would tend to be inherited together. There are different types of linkages according to, on the basis of different criteria. So on the basis of the crossing over occurring in linkage, or on the basis of genes involved, And the chromosomes involved, there are three types of linkage group. Linkages are three types of linkages. First is the based on the crossing over, it will be complete linkage or incomplete linkage. So the complete linkage, it is in case of males of Drosophila or females of silkworm, there is a complete absence of any recombination type due to the absence of crossing over. So there is no, as there is no crossing over occurring, there is the all the genes which are present in a linkage group, they will transmit from one generation to another generation completely. So there is no chance of any crossing over. So this is called as the complete linkage. While in case of incomplete or partial linkage, there is some frequency of crossing over is there between the linked genes, and it is called as the incomplete linkage. It is occurred in case of maize, pea, and several other organisms. Then second type of linkage, it is based on the genes involved, whether the genes are in a dominant position or they are in a recessive positions. So accordingly, there is a coupling phase and repulsion phase of linkage.
Yes, sir.
Hello. Hello.
Yes, sir. किती slides आहेत अजून बाकी?
आणि मी 2 मिनिटात संपवतो, काय प्रॉब्लेम नाही. There is coupling and a repulsion phase. So if the both alleles are dominant in nature, this is called as the coupling, and when one dominant and one recessive is present, this is called as the repulsion phase. And the third type is based on chromosome involved. If the genes are located on other than sex chromosome, so it is called autosomal linkage, and then it is a sex linkage in other type. So this is having some linkage, is having some breeding significance in breeding. So it is undesirable when the desirable and undesirable genes are linked together, but it is desirable when the two desirable characters are linked together. So the breeder can select one and automatically simultaneously will improve the other character. So this is all about linkage. So thank you very much, Male sir and Dr. Devkar sir, for giving me the opportunity to share my some classes with your JRF students. And if in future, if some student is very much interested, he may ask or he may contact me for discussing some of the topic which he finds that it will be very difficult to understand from the books. So thank you, sir.
Thank you, sir. Thank you, sir. Thank you for very nice lecture, sir. Thank you.
It's my pleasure, sir. Thank you, sir. Thank you very much.
So I would like to invite Dr. Kabre sir to introduce our next expert. So please, Kabre sir.
Good afternoon, friends. So friends, in this our series of lectures for your JRF, SRF examination, today we are having a very eminent personality, Dr. D.S. Pokharkar sir. Actually, he is our head. He was our head department of entomology, NPKV Rauri. He has a vast experience in the field of this biological control. He was at Pune also. So we are just getting a very good inputs from him. Pokharkar sir, I welcome you, sir, here on this platform, and I am just requesting you, because he is now retired, he is having his own schedule, but in spite of that, he has agreed to deliver the talk on this topic. Shinde sir, तुमचं हे screen तुम्ही हे करा sir. Shinde sir, screen sharing तुमचं हे करा. Okay, thank you. So I request Pokharkar sir, welcome here on the behalf of our College of Agriculture Dhule and Mahatma Phule. I welcome you, sir, and thank you very much for accepting our invitation to deliver a talk on this biological control. Okay, sir.
Okay, sir. Okay, thank you. Thank you. Hey, my video down thoda. PPT कुठे आहे?
स्क्रीन शेअर करायची आहे
का? हॅलो महाले सर.
Yes, sir. Yes, sir.
हे स्क्रीन शेअर करू का?
हो सर. हो.
सुरू करायचं लगेच का? एक
2
मिनिटं ब्रेक घ्यायचा आहे.
नाही करा सर, काय हरकत
ओके
नाही. Con-
ओके ओके. Hello, good afternoon, everybody. Respected Dr. C.D. Devkar sir, then my colleagues, Dr. Kabre sir, Mahale sir. So, as you have given opportunity to speak for the JRF SRF continuous lecture series, I am going to deliver the lecture particularly on biological control. Okay. So here, first let me introduce what is biological control. So in the discipline of entomology, we are going to study the insects in all respect, useful insects, harmful insects. So under the harmful insects category, the insects, those which can cause harm, particularly to the crop plants. So which is of course a interest of the farmer. So we regard them as a insect pest. So here in this particular case, how to control or how to make use of these different strategies for the control or reduction of this particular pest, so that we regard as a pest control methods. So here under these circumstances, 6-7 pest control measures are there. We know this: cultural practice or cultural control, mechanical control, biological control, chemical control, quarantine methods. So such different methods are there that we are going to utilize artificially, and then we are going to reduce the pest population density. And so that's why accordingly we can get the economic control. So in this particular case, so biological control method, here we are going to utilize any living organism. It may be insects, it may be other than this insect, that we can say non-insects. So we are going to utilize these particular organisms. These organisms, they are known as natural enemies, isn't it? So here these natural enemies we are going to utilize particularly for the control of insect pest, isn't it? So that's why, so when primarily we say what is biological control, it is a method of pest control that employs particularly parasites, parasitoids, predators, entomopathogens, these natural enemies, to reduce down the pest population. That particular pest population is reduced and below economic injury level. You must know, as a student of this agriculture and particularly entomology, economic injury level, economic threshold level, and economic damage. These three levels, they are of course more important in maintaining the pest population density. Here, when the pest population goes beyond economic injury level, then we regard them as a serious pest, and then we undertake such a sort of artificial pest control strategies. So here in nature, these natural enemies, parasites, predators, pathogens, they are available in nature. So it is not at all separately synthesized or artificially synthesized. They are available in nature. Only we are going to identify, recognize, we can mass culture them and utilize for the control of insects. And that's why they are also known as the bioagents. So these particular bioagents, they are going to take care of the pest. So here in this particular case, in the pest management strategy, after 1959, Stern et al. He put forth such an idea. Instead of separately using these methods of pest control, all methods, different cultural, mechanical, biological, chemical, or these other tactics, utilize them separately. Instead of using separately, we can combine them and make a pest management strategy. That is, we regard them as a IPM tactics. So here in this pest management tactics involving purposeful application or purposeful use of natural enemies or manipulate natural enemies to reduce down the pest status, that is pest population below economic injury level, that is known as biological control. Usually this Paul Dirac, you might have heard the name because he is basically a very great eminent scientist. In 1964 he wrote a book, Biological Control of Insect, Pest and Mites. You note down this particular, this is one of the important question it will ask. So this particular book, it is basically written and compiled by Paul Dirac in 1964, and he has given it all these basic ideas from ecological point of view, how these organisms, parasites, predators and pathogens, how they are utilized for the control of insect pests through introduction, conservation and augmentation techniques. So that's why the basically he has given this definition, that is third definition here it is given, the action of parasites, predators and pathogens in maintaining this another organisms population, that is insect population, at lower average than would occur in their absence. So this is how this basic idea it is there. So what are these biocontrol agents or natural enemies? As they are naturally available, that's why we say natural. And they are going to destroy the insects or pest particularly. That's why we regard them as a enemies for the pest, that is natural enemies. And so as they are living one, so that's why we regard them as a biocontrol agents. So here in this particular case, first one, that is parasites. So here I have simply mentioned that is parasitoids, but basically that term it is a parasite. Parasite means one organism living in association with other organisms and feeding on and developing its life and up to the maturity stage. So it is known as a parasite. Very simple example it is there in there in this particularly one, the such a sort of these organisms, it may be of any kind, it may be of any phylum, it may be of any taxon, so that we regard them. Sometimes these animals for example, we are observing these higher end cows, buffaloes are there, and these ticks they are available. The ticks they are living in association with animals and sucking the blood from them. It is regarded as a parasite because animals they are belonging to chordata and the ticks they are belonging to another group. So here that type of this category it is there. That means one organism living on others, irrespective of not only regarded on the basis of taxonomy, any kind of taxonomic nature. Another example it is there in human. There is a body louse, it is there. The louse, it is an insect, but human being, it is a chordate animal. So here in that particular case, body louse, it is considered as a parasite. But as far as this entomology is considered, we regard them, we utilize this term as a parasitoid. So try to understand what is parasitoid. Parasitoid means it is an insect or parasitizing or living in association with other arthropods, which is taxonomically a insect nature. Insect, that means one insect living in or on or in the association with other insects and devouring or obtaining their food for its growth and development and complete its the life. So that's why it is regarded as a parasitoid. So that in that particular case, parasites and parasitoids, they are of course similar terms, but parasitoid is particularly used for the insects, those which are parasitizing insect, that means similar taxonomic nature. Another group it is their predators. Predators, that means this organism, which is of course bigger in size, which kill and devour the prey promptly. Very simple example in this chordates I will give you. We can take the example of these cats and rats. Cat is a predator, while these rats, it is a prey, isn't it? Very simple example. In the similar case, in entomology also, some ladybird beetles are there. They are going to prey upon these aphids. Aphid is a prey, the ladybird is a predator. Understand? Then the third one, that is pathogens. Pathogens, generally we are going to observe in nature bacteria, viruses, fungus, protozoa, rickettsia, mycoplasma, so many these microbes they are there. So here in this particular case, these microorganisms we select specifically such microorganisms which are pathogenic or causes infection in the insects particularly, so that they are regarded as entomopathogens. Then this fourth category, it is their non-insect. Non-insect, that means other than this arthropod group. This some of these birds, some of these other animals, some of these frogs, lizards, they are going to feed upon insects. So that is known as a non-insect organisms. And these predatory vertebrates. So likewise, in 4 or 5 categories are there. So accordingly, we see one by one. This first one, that is important organisms in the biological control science, that is parasites or parasitoids. I already told what is the critical difference between parasites and parasitoids. So here in the entomology, particularly we are going to utilize the insects for the control of insects, that is parasitoids. Insects that parasitize other insects and these particular arthropods, they are called as a parasitoid. So this is very important point, that to distinguish parasites and parasitoid. So here parasite is an parasitic organism, parasitic insect which develop in the body or on the body on a single host or from, and they are going to feed upon any stage of the insect. Either in the insects you may find there are 4 stages, complete metamorphosis, 4 stages are there: egg, larva, pupa and adults, isn't it? So here some insects they parasitize only egg stage, some insects they parasitize only larval stage, some insects they parasitize only pupal stage. So accordingly, egg parasitoid, larval parasitoid, pupal parasitoid, they are named. In case of incomplete metamorphosis, there are 3 stages: egg, nymph and adult. Is here in that particular case, egg stage or nymphal stage, they are parasitized, or nymphal and adult stages, they are parasitized. So accordingly, these particular insects, they are named as a nymphal parasitoids or nymphal adult parasitoids. Then let us see, as far as the topic is considered, there are several kinds of parasitoids depending upon the stage of the insect attacked. So we are going to categorize them as a egg parasitoid. So very best example of this egg parasitoid is Trichogramma chilonis. You keep it in mind. Trichogramma chilonis, it is the egg parasite of the most of these lepidopterous insects. So here in that particular case, you will find in case of the stem borers, the jawar stem borer, maize stem borers, so sugarcane stem borers, we are going to utilize, or even only the bolworms in cotton, we are utilizing Trichogramma chilonis as an egg parasite. Even in case of this okra, okra fruit and shoot borer, brinjal fruit and shoot borer, tomato fruit borer, here we are going to utilize Trichogramma to parasitize the egg stage. That means when these insects they lay their eggs on the plant surface, these trichogrammatids lay their eggs in its host eggs and destroy the egg in the adults, in the egg stage only. So that's why there is no larval stage to cause damage. So another important species is Trichogramma japonicum. Trichogramma japonicum, it is a such a sort of the species which is mainly used for paddy stem borer, even this top shoot borer in sugarcane. Because here these particular insects, they have the egg mass, it is covered with scales. So that's why these trichogramma species, it has long ovipositor to deposit the eggs. So besides that, this trichogramma evanescence, it is there, trichogramma, then pretiosum, it is there, trichogramma brasilensis, it is there. Near about these 20 important species of these trichogrammatids, it is there. Depending upon this type of these insects and host plants, they have association with the egg stage of the host. Second parasite, it is their egg larval parasitoid. Here egg larval parasitoid, that means this particular parasite, for example Chilonus blackburni, it is going to parasitize the egg stage of these insects and come out as an adult from the larval stage. So that's why two stages of the insect, so it is required for its growth and development, that is egg and larvae of the host. Example is potato tuber moth. Potato tuber moth, it is controlled by this Chilonus blackburni. Then this pink bollworm, it is controlled by this particular insect. So here this Chilonus blackburni, it is a uniparental parasitoid. Uniparental, that means there are no separate males and females. A single individual can perform the characteristics of both male and female. That's why it is known as uniparental. It is the most versatile parasitoid. Besides that, another example it is there of the egg larval parasitoid, Copidosoma cohileri. Copidosoma cohileri, as at our this Pune college, we have this particular such a sort of this AICRP center where a lot of work on potato tuber moth it is done. Near about this 20 years I worked there, and we have given this particular release this particular parasitoid Copidosoma cohileri for the control of potato tuber moth. Then this another larval parasitoid. So these particular here, this Apentele species and this Bracon species. So these are the larval parasites. These adult stages of this particular insect, they find the larvae of this Lepidoptera, that is moths and butterflies, parasitize them and complete their development within the larval stage of the host. Bracon inhibitor, it is one of the most versatile and indigenous parasitoid, it is there. Appenteles glomeratus, it is also a good parasite, it is there. Then this Appenteles plutelli, or we can say Cotesia plutelli, it's a name, it has been changed. So it is one of the good larval parasitoid for diamondback moth in cabbage. Another one, that is Campholetis chloridi. So this is a larval parasite of Helicoherpa armigera, particularly in cotton ecosystem or wherever this pigeon pea, chickpea, or even in the case of this tomato, this Campholetis chloridi, natural population, it is there abundantly, and it is going to parasitize the larvae of Helicoherpa armigera. Then Irivorus trochanter, it is also a larval parasitoid. So likewise, we can see such a sort of these particular examples, and even you will find in this second picture, you will find such a sort of this several this whitish pupae. It has been observed. It is a parasite pupae coming out from this host larvae. Then this larval pupal parasites. Here, these particular tachinid fly. So dipteran parasite, it is there. So it is going to parasitize the larval stage of the insect and come out from during the pupal stage of the prey, in pupal stage of the host insect. So it is also one of the important potential as well as indigenous parasitoid, it is there. Then this, in case of incomplete metamorphosis, wherever this egg, nymph, and adult stages are there, there we find this nymphal adult parasitoids. Here, Aphelinus mali, it is one of the important and successful example in India, particularly for aphids. Then Encarsia formosa, Encarsia formosa, it is a nymphal and adult parasite, important nymphal and adult parasite of white flies. So here in this particular case, in Maharashtra, or particularly in India, this we find this in last decade, spiraling white fly, it was introduced, particularly in peninsular region. In that particular case, we introduced Encarsia gaudolopi. So here, this particular, it is a very potential parasite for control of spiraling whitefly. Then another successful example, it is there, sugarcane woolly aphid. Sugarcane, in case of sugarcane woolly aphid from 2002 to 2006-7. So in the last decade, sugarcane woolly aphid, it was a very serious pest, and by that time we introduced, as our Pune center has introduced, Encarsia flavoscutelum, this nymphal and adult parasite, particularly for this pest. Then this Epiricanium melanoleuca. Epiricanium melanoleuca, it is a Lepidopterus parasite, and particularly for the sugarcane pyrilla. Nymphal and adult stages of this particular pyrilla, it is parasitized by the Epiricanium melanoleuca. Then these important features, important examples I have given. Besides that, in the last decade, we have also find that this Acerophagus papai, it is one of the important parasitoid, and it has become a classical example for this biological control of papaya malleable. That our Pune center, they have introduced, they have identified and gave a support, particularly in suppressing the pest population, mealybug population in papaya, particularly in India. Then this another important organism, that is predators. So I will give you the very simple example. For example, you will find in the jungle, forest ecosystem, so many animals are there. Herbivores are there, carnivores are there. Usually, among these carnivores, you will find these tigers, lions. They are the predators. And while small other organisms, small other animals are there, they are the prey, isn't it? So here, accordingly, if you take down, consider this particular example, then you will come to know the idea of this particular, yes, the idea of these predators. Another simple example it is there, that is cats and rats. In the early history, it was noted that these Egyptian ladies, they are going to rear the cats, particularly in the houses, to control the rats, isn't it? This is one of these ancient example of biological control. So that it has been given. In 1962, another example it has been given, particularly this Indian myna birds, Acridotheres tristis. These particular birds, Indian myna birds, it has been utilized for the control of locust. And so the Mauritius government, they have imported these myna birds from India to Mauritius for the control of locust. So this is how such a sort of this other than this insect, these other examples of the predators are there. So you will come to know idea what is meant by predator. It is a free living organism which kill its host, that is prey, immediately or directly by attack, and feed them promptly. It requires a number of prey individuals to reach maturity. For example, if you find such a sort, a cat today, if it feeds on a single rat, on the next day or 2 days later, it requires again another rat to kill it, because it is a food. So that's why number of many rats it has been required for the control, for the development of such a sort of the prey. So here, according to this such a sort of this, particularly in this arthropods, different orders are there, and in different orders, you will find different kinds of the predators are there. In beetles, Coleoptera particularly, you will find coccinellids is the best example for indigenous predators. You might have heard the name ladybird beetles. Ladybird beetles, wherever you will find this lot of aphid population, it is there, particularly in this vegetable crops or even in this peas, pulses, you will find this, they are abundant. Here it is known as Coccinella septum punctata. So these are Coccinella rependa. So here these particular species, they are indigenous in nature. Indigenous, that means they are available or they are originated from India, isn't it? The species which are available or originated from India, it is known as indigenous. The species which are imported or introduced from the other countries, it is known as exotic. So here this ladybird beetles, another species it is there, that is Minochilus sex maculata. Here the second picture it shows, that is Minochilus sex maculata or Chilomenus sex maculata. It is also a predator found particularly on the aphids, scales or soft-bodied organism, that is nymphal and adult stages of scales and mealybugs. Then this Vedalia beetle. So Vedalia beetle, it is the first successful example of biological control. So here Rhodolia cardinalis, it was utilized for the control of cottony cushion scales, particularly in this California, the citrus industry, it was damaged. And so by that time, this United States Department of Agriculture, they have imported this particular Vedalia beetle or Rhodelia cardinalis from Australia to California, and they have saved the citrus industry. So that's why this is one of the first successful example of biological control. You keep in mind. Then this Australian ladybird beetle. So here in the last 2-3 decades, we are utilizing this Cryptolemus montroseri, particularly in grape vineyards, for the control of grape mealybugs, Maconellicoccus hirsutus. In this grape industry, usually what happens, because of continuous spray schedule, this mealybug population, it has been research. It has been become a very serious and other pests become minor. So by that time, these mealybugs, it is very difficult to control because they have waxy coating on its body, and so under any circumstances, chemical pesticides they cannot control, or they may not provide such sort of desirable pest control. So that's why here this Cryptolemus montroseri, first time in 1988, it was introduced in India, particularly in South India, and now it has been established very well, particularly in the grape industries or even in this orchard ecosystem for the control of mealybug population. Then this another ground beetle, it is there, carabids. So these are the general indigenous predators, but they are polyphagous. They are not so specific about the host, but simply they can find such a sort of any larvae or any such a sort of organisms. It is a general predator. Then this chrysopa. You might have heard this Chrysoperla carnea. After this ladybird beetle, second and important predator, particularly in India, it is nothing but the Chrysoperla carnea. Another species, that is Chrysoperla celestus, it is there. So it belongs to the order Neuroptera. So these particular insects, they are these adult stages as well as larval stage. First picture, it is nothing but the larval stage, and the another is adult stage. Both these stages, this larval stage as well as adult stages, they are feeding on all soft-bodied insects, aphids, jacids, thrips, mites, or even the eggs of this Lepidoptera, that is bollworms, etc. So this is one of the very potential predator, particularly for the control of insect pest. Then this, in case in Hemiptera order also, we are going to utilize. These are all indigenous, they are available. Pentatomid, that is seed bug, or even reduvid bug, or it is also known as assassin bugs. This Rhinocoris piscipes, which is going to feed upon the eggs as well as these larval stages of the Helicoverpa armigera, Platimeris levicolis. So these are merely examples are there, but they are not that much specific. That's why we are not commercially utilizing. Myriad bugs it is there, Septorhinus levidipenis, which is available in the rice ecosystem, or Velidae, that is Microvelia ultralineata, which is also a in the rice ecosystem, particularly feeding on rice spa. Then this dipteran predators are there. So here I already told that is syrphid flies. Syrphid flies are hover flies. It is just like a house fly, but it is bigger in size. And so these hover flies, they lay their eggs in these aphid colonies or jacid colonies, and after hatching their eggs, their maggots, they start feeding on these aphids. Aphid, that means these maggots, they suck the body sap from the aphids, they shrivel, and they are going to reduce down the population. Then robber flies. So these are all indigenous predators are there. So they are not that much potential, but they are general one. Then this another category, that is mantles, Mantoli family. So here praying mantles, it is also a general predator. So and the next one, that is Odonata, the dragonflies and damselflies. So these particular dragonflies and damselflies, they fly in the air. Most fast flying insects in nature is a dragonfly. So these dragonflies, during their flight, they capture small living organisms and feed upon them. While their larval stages, they feed upon these larval stages of these dragonflies, they are in the what? Aquatic. They feed on mosquito larvae. So that's how they are going to be a versatile predators. So likewise, parasites and predators, these are the insects. These are living organisms, and they are going to utilize for the control of insect pests. So this is how these two agents, they are important. Besides that, these pathogenic organisms, bacteria, fungus, virus, protozoa, rickettsia, they have also potential to cause diseases, particularly in the insects. But here, what happened? So these organisms in general, they are also infectious to plants. They are also infectious to other higher organisms. But in this particular case, when we are searching for these entomopathogens particularly, we are going to isolate such organisms from dead insects, which are very specific, particularly for that particular group of insects. First organism is a bacteria. So here, this particular bacterium, it is going to cause disease conditions, particularly in the Lepidoptera as well as Coleoptera. Here, Bacillus thuringiensis, we regard them as a BT, isn't it? Bacillus thuringiensis, it is one of the important this bacteria which produces delta endotoxins, and it is going to cause particularly infection in the insects. While Bacillus popili, it is particularly causing a infection in the Lepidoptera, that is particularly Japanese beetle, Papilio japonicum, or even in case of this, the such a sort of this white grubs also in some of the cases, you will find this infection. Here, this Bacillus popili, it is a obligate pathogenic bacteria. That means they always cause infection where the insect they are living, and it is going to kill it. While this Bacillus thuringiensis, it is a facultative group, or it for culturing it, it requires this bacterial medium, not necessarily this host insect is required. So here you will find the first picture it is given, that is bacterial infection in the Lepidopterus insect. After this infection, when these insects they are feeding on the plant surface, automatically some bacteria, it will be consumed, it will be enter into the digestive tract. So then and then it will cause infection. So that means this Bacillus thuringiensis or Bacillus populi, this bacterium, it must be ingested by the insect. It must be eaten up. So that's why it will enter into this foregut or gut, then these particular bacteria, they release delta endotoxins and causes infection, isn't it? So here similar is the case with Bacillus populi. Bacillus thuringiensis, it is available in the commercial form, Delphine or Dipel, the Halt. So many formulations, they are readily available in the nature. Then this second organism, it is there, that is fungus. So these particular bacteria, they are going to cause infection. Sorry, this fungus, they are going to cause infection, particularly in the larval stages of the organisms. So here in this particular case, the larval stage, which is having a soft body, and so that's why these particular mycelium or conidia, they will incorporate or introduce into this body, and they are going to cause infection. So it is just like a contact poison. It is entered through the cuticle. So it need not to be ingested. It should not be eaten up by the insect. So that's why when it will come in contact, when this the fungal population or this fungal culture, it is available on the plant surface and insect is going to move. So by that time, this particular the spores or this conidia, it will get attached to the insect body and automatically it is going to develop. So here this first example, it is there, that is Metarhizium amisopli, which is a successful example. It is also known as green muscardine fungus. Then the second example, that is Beauveria bassiana. So this is a white muscardine fungus. Third, then Pneumonia relay. So here, besides that Verticillium licani, it is also one of the important this fungal pathogen, which is going to cause infection, particularly in the soft-bodied insect like aphids, jacids, thrips, mites, etc. Then the third potential example, it is there, that is viruses. There are 5 to 6 groups of viruses are there, which is pathogenic to insects. But amongst them, baculovirus. Baculovirus is most infectious. So under this baculovirus, NPV and granulosis virus, it is there. So here I have quoted only one successful example that is there, that is HNPV. HNPV, that means Helicoverpa armigera nuclear polyhedrosis virus. It is infectious to Helicoverpa armigera. Then SLNPV, Spodoptera lichura NPV. Besides that, granulosis virus, which is available particularly in early shoot borer in sugarcane, in potato tuber moth, diamondback moth. So these are very infectious to the insects. Then this protozoa. Protozoa, it is going to cause a infection, particularly in locust, Bombay locust. So that's why Nosema locusti, this particular species, it is infectious, but it does not have that much potential to suppress the pest population. And this fifth organism, that is Rickettsia. Rickettsia, it is also going to cause infection in the insects, but to kill the insects, it requires near about 40 to 50 days. And so that's why the insect has been already reached to the from egg stage to adult stage. So that's why they are going to cause infection, but it is not having that much potential to utilize them as a biocontrol agents. Then the nematodes. In the non-insect nematodes, Neoplectana carpocapsi. So DD136. This type of the nematode, it is going to cause infection to the insects. So this is one of the successful example. Then these mites are there. Mites, so here in the insects, red mites are there, two-spotted mites are there. They are feeding upon the plants. But some other predatory mites are there. Amblysia species or these particularly predatory mites are there. They are feeding on the other species of these predators. And this third non-insect pest is non-insect biogen, that is spiders. Spider, it is a general predator. You will find in the middle picture these particular predators. They are feeding on all stages of the insect, maybe egg stage, larval stage, pupal stage, or adult stage. Then this vertebrates. So invertebrates, you will find the predatory birds are there, reptiles are there. You will find the snakes, lizards. You will find we are very much afraid of the snakes. So isn't it? But these particular snakes, they are going to feed upon the some other organisms. They are, so that's why they are also considered as a good predators. Lizards, we will find them particularly in houses, dark places that to feed upon insects. So besides that, your birds are there. So the birds, they are going to also feeding upon different stages. When you will find in the off season plowing, it is going on. So many birds, they are running behind the tractors. So by that time, the subterranean insects, when they are exposed, so likewise the birds, they catch and they feed upon them. Amphibians, frogs and toads, particularly in the huh, hello. Then we will see some important successful examples of biological control. So already I told you that
आवाज कमी आहे.
Fedalia beetle, Rhodolia cardinalis, it is one of the first successful example which is introduced into California and later on even in the Egypt and also in India we have introduced from the Australia for the control of cottony cushion scale Iceria purchasing. So this is one of the always it has been commonly asked questions, particularly in MCQs. So you will find these particular pictures. Then this Aphelinus mali. This is a very specific predator, particularly for the woolly apple aphids, Eriosoma lanigerum. So these particular in Himachal Pradesh or even in this Uttaranchal, it has been introduced particularly for the control of aphids, woolly apple aphids, particularly in the apple ecosystem. Then this Telonomus remus, this particular parasitoid, it has been introduced particularly for the castor semilupus, Acajanata. Then the predatory Platymeris flavicolis, it has been introduced from Zanzibar for the control of this coconut rhinoceros beetle. Then this some examples, particularly even in the case weeds are there. Weed, it is also regarded as a pest. So in that particular case, you will find earlier near this you will find cactus. It was spread in these forest areas or even in this mountain hilly areas wherever this it is not going to allow to develop the grass, and so that's why these shepherds have a problem. So that's why in order to control this particular cactus prickly pear, so here we have introduced this cochineal insect Dactylopius tomentosus, particularly from Sri Lanka, and we have released. It is just like a white mealybugs we will find. So these insects, they are feeding upon this cactus, and now at present you will find this in this mountainous area. This cactus is not become a pest; it become a classical example of biological control. Then this another weed, that is Parthenium weed. Parthenium weed is introduced in India in 1970s. So from USA. So in that particular case, it has been spread all over India and occupied barren as well as even agricultural lands. So in order to have this undertake this Parthenium eradication campaigns, government of India, they have utilized many schemes, many projects spent a lot of money for this control of this particular pest. But we have introduced this Zygogramma bicolorata as a Mexican beetle. So you will find these pictures at the upper right corner. These beetles, they are feeding on these all stages, flowering stages, here, even on the leaves, even birds, and it is going to destroy. It is a slow feeder, but it become a very successful example of biological control of Congress weed. Then this water hyacinth. Third, you will find the jalpardi. Water hyacinth, Echornia crassipes, it become a aquatic weed. So here, particularly in South India, in Karnataka, Kerala, you will find this these most of these ponds or these water bodies, or even in Maharashtra also, the river beds or dams, ponds, they are covered with this the water hyacinth. So here, every year to eradicate them mechanically, it is very difficult. So that's why we introduce this Neochatina echorni and Neochatina bruchi. Two species are there. These curculinid coleopteran weevils are there in Kerala as well as Karnataka. We have also introduced in Maharashtra through our this biological control Pune Center. So these three important biological control of weeds, they are of course a successful examples of the biological control. So here, how to utilize these biological agents, isn't it? How to utilize these parasites, predators, or microorganisms. So there is some techniques or principles are there. So in these basic books, it has been given that is introduction, conservation, and augmentation. Introduction, that means wherever these particular introduction that which is resembles with that of this inoculation, wherever such organisms, that means maybe parasites, maybe predators, or maybe microorganisms, they are available in any way, any part of the state, any part of the country, any part of the world, you import them, try to culture them under laboratory conditions. That is known as mass culturing. And frequently during the season, you can introduce, you can release them. So that is known as introduction. So here in that particular case, naturally they are going to spread in the nature, and they are going to perpetuate by air, or they may run behind their host or prey, and they are going to take care. Second one, that is inundation. Here this conservation is more important. Conservation, that means these the whatever indigenous natural enemies are there, or whatever introduced natural enemies are there, we have to protect them from these natural calamities or adverse conditions. And you can supplant, you can make support them to develop or establish under field condition, that is known as conservation. So here this inundation, second step it has been given, inundation, that means frequent releases. Whatever this mass culturing we undertake under laboratory conditions at weekly interval, for example, trichogramma, at weekly interval or 7 to 10 days interval, we have to release them under field condition, that is known as inundation. And third one, it is known as augmentation. Whatever these natural enemies we have introduced, we have conserved, when we have to augment, when their population under field condition is not sufficient to control the pest, we have to add them into the nature. That means we can make a inoculative releases under field conditions. When there are five ladybird beetles in a 1 square meter area, and aphid population is extra, we can release 10 more so that they can finish the pest population very readily. So this is how this introduction, conservation, and augmentation, these three methods or three techniques of pest controls are there. So here in this particular case, the basic difference, you must know these parasitides and predators. So here usually in the definition itself, we have given parasitides, they derive their food from their host regularly, because parasite is always attached with the single host, and it is going to feed upon a single host and develop them. While the predators, they feed upon many of the hosts. Many. That means today this ladybird beetle has taken up these 5-10 aphids in the morning. Again in the afternoon, again 5-10 aphids it is there. So that's why these predators, they catch, kill, and devour the prey promptly. The parasite, second, the distinction it is there. Parasite remains close in association with their host. Always it remains. Parasites always remain either inside the body or on the host of the insects. While the predators, they live independently. For example, you will find the cats, they are moving independently. They are not mixing with the rats. So likewise, wherever they find predators, they find hunger. Accordingly, they find their prey and they can mm, undertake such a sort of this control of the prey. Third, it is there, that is parasitoids are usually smaller. The parasitoids are usually smaller than their host. On the contrast, predators are always bigger than the host because it has to fight, isn't it? Always you will find ladybird beetle is bigger than the aphids, isn't it? Then this fourth, it is there, parasites, they are sluggish. Sluggish, that means they are very small, microscopic or submicroscopic, and they always remain attached with these insects or in the colony of the insects, and they feed upon it. While predators, they have larger, and they have long life cycle. Then the parasites complete their life cycle within mm, complete their life cycle earlier or before the host may die, while predators have longer life cycles. Sixth, it is there, parasitoids generally need a single host, single insect to complete their life cycle, while the predators, they need many individuals to feed daily throughout their long life. Host determines habitat of the parasite. So wherever the insect is there, the parasite has to stay there. On the contrary, here it is the prey never determine the habitat of the predator. The predator live independently, prey live independently. So likewise, 6-7 differences are there. Examples are there. I already told trichogramma, apentales, bracon. These are the examples. While under predators, we will find ladybird beetles and Chrysoprula. Then these desirable attributes. What are the important characters? These bioagents, how it should be selected or how far they are utilized. For example, they must have this such a sort of this 8-10 characters. These parasites, predators, they must search their prey. That is high searching ability, high host searching ability, it should be there. The insect will not come to the predator and it will say, no, I am your prey, you may eat it. It is not like that. So it should have high searching ability to find their host. Then host specificity. So, for example, the ladybird beetles, they must feed upon these aphids, jacids, soft body. They should not switch over to the other insect. Egg parasite like trichogramma, they must parasitize only these Lepidopterus eggs, Lepidopterus insect legs, not other. So that's why the potential of that parasite will be reduced. It should be monophagous and not never polyphagous. In the third one, it should have short life cycle and high fecundity. That means it must breed fast than the host. Then its mass production must be easy, so that we can undertake under laboratory conditions. Its life cycle should be matched with the host insects. That means egg parasite should breed with the egg stage in coinciding with this egg stage. That means trichogramma adult stage should be coincided with the egg stage of the Lepidoptera. Adaptable to new environment. It must ecological adaptations it is there. It must have quick dispersal power. Then this it should be always such a sort of the enemies under any circumstances they should not feed upon plants. Parasites, predators, or pathogens. They should not be pathogenic or they should not herbivores. Then this hyperparasitism. Hyperparasitism, it is one of the negative attribute which is going to withdraw or lessen the potential of the parasites and predators. So that's why hyperparasitism should not be observed in any parasites or predators. And it should stand under refrigeration, because when we undertake mass production, we should at least store at 8 degrees centigrade temperature under refrigeration. So this is how some desirable attributes are there. Then we will see its advantages and disadvantages.
Okay, sir. In 2 minutes, you can conclude, sir. Okay,
Yes,
sir?
yes, yes. Within a minute, one or two.
Yes.
So you will find what are the advantages? Biological agents. Biological agents, they are living organisms. So they are self-propagating and they are self-perpetuating. That means parasites and predators, they undertake their own production. They can produce themselves under laboratory conditions and they can migrate. That is known as self-perpetuating. While if you apply the chemical insecticide, it is a physical poison. It is not run behind this insect. Wherever it is there, it is there. Then this no harmful effect to man and other organisms. So that's why such organisms have been selected. Then this third important one, that is once biological control has been established, it is a permanent control. It is not a, for example, in the last decade, we undertake biological control of sugarcane woolly aphid, Dipa aphidiflora and Micromus aeroratus, and this Encarsia flavoscutullum. These three organisms have taken care these sugarcane woolly aphids in the last decade. In this early period of the decade, this Acerophagus papayi, it has taken care this papaya mealybug. These are the recent examples. It is there, you keep in mind. No resistance. Resistance is never developed. It is unlike to the insects. No resurgence or secondary pest outbreaks. Once parasites, predators have established, they won't allow this parasite, this insect pest to develop further. And it is economical. Within a very short period or this low investment, we can undertake the production. And it is compatible with other methods except chemical. So these are some important attributes, important advantages are there. While this besides that, the disadvantages are there. It has narrow host range because host is specific. One organism will kill one parasite. For example, Copidosoma coileri will take care of this potato tuber moth. No adults, no these other insects. Then it is a slow feeding, so that's why it will require some long time. So if we are going to take this, release this trichogramma, its this its repercussions or effects will be noticed after this 10-15 days, because it is going, if you utilize this NPV, so in larval stages will be there. After 1 week, disease larvae will be there. That means it is slow acting. Then limits the use of pesticides for other pests. So it happens. Another high trained manpower it is required for bioagent rearing, because it is a technical work. It is there difficult to maintain quality of bioagents. There should not, because sometimes in the parasites, predators, continuous this inbreeding, it may descend down the cultures, or even this continuous this culturing, subculturing, it makes this it reduces the virulence of the organism. So that's why that particular conditions we have to always take into account, and specific environment required. Here this, for example, fungal pathogen, it is there, it requires high humid conditions. They have less potential, particularly when this temperature goes beyond 30-35 degrees centigrade. So this is how these biogens or biopesticides, they have very good potential in the biological control of insect pest.
Okay. Thank you very much, sir. Please,
Okay, thank you, sir.
please stop sharing, sir.
Oh, yes, yes,
Okay, sir.
yes.
On the behalf of of our College of Agriculture, Dhule. I am very much
Okay, sir.
thankful to Dr. D.S. Kokharkar, sir. His very schedule and so many things. He has given a very nice information. Actually, he is a boss in our biological control. He was in charge of AICRP biological control also. So thank you, sir, for
Okay.
giving very good information to our students regarding this biological control.
Okay, thank you. Thank you. Aditya?
Now,
He's sharing.
we have for another lecture, we have with us Dr. Kshirsagar, sir. Sir, I welcome you, sir, on the behalf of College of Agriculture, Dhule, on this platform where we are conducting the classes for JRF and SRF examination. Dr. Kshirsagar, sir, is the Associate Professor of Horticulture at PGI, Postgraduate Institute, MTKV, Rahuri, and he is also having vast experience in almost all the fields there, because he is in PG, so naturally he has studied all the things. So we have a very good resource person today to deliver this talk. So once again, I welcome you, sir, Dr. Kshirsagar, sir, and you can share your screen for your presentation.
Thank you, sir. Thank you, sir. I am audible.
Up to 5 o'clock, sir. Okay.
Okay, no problem, sir. I am audible. Okay.
Okay, thank you, sir. I
am audible.
Yes, you are audible, sir.
Okay, I will share this.
Okay,
So
sir.
thank you, respected Dr. C.D. Devkar, sir, Usher Dean, College of Agriculture, Dhule, all the staff, College of Agriculture, Dhule, and my colleague, Dr. Kabra, sir. So I welcome all of you, all the students here, so that I will share my experience and lecture on vegetable breeding, methods of breeding of vegetable crops. So I'll share the slides. So can you see the slides? Hello. Yes. Okay. So our topic is breeding methods in vegetable crops. So first we'll see what is the vegetable breeding. You know, vegetable is one of the important sector in horticulture, is playing very important role. So what is the vegetable breeding? So first we have to understand it is concerned with the modification of genotype of the plant. You have to go for the modification of genotype of the plant so that they become more useful to the humans. That is the first priority in this vegetable breeding. And this depends upon the reproduction system of the plants, whether the plants are self-pollinated or cross-pollinated. So that is very important when we are using any breeding system. So whether it is a self-pollinated or cross-pollinated. And main objective of the vegetable breeding is to develop varieties which are superior to existing one. What are the different objectives? Why we have to go for the breeding? So that is very important. This vegetable breeding is the art and science of changing and improving the heredity of the vegetable crops. So here, what type of plants are there? Self-pollinated vegetable crops or cross-pollinated? There are different breeding methods here. So self-pollinated crops, tomato, okra, lettuce, brinjal, chilies, potatoes. These are all beans, peas and beans are also comes under these self-pollinated crops. Cross-pollinated crop, all cruciferous and cucurbits crops are cross-pollinated, cabbage, carrot, cauliflower, then watermelon, muskmelon, so many crops comes under all these So breeding methods for cell-pollinated crops. So what is the vegetable breeding? Why we have to go for this breeding? So breeding is nothing, it is the art and science of changing and improving the heredity of the vegetable crops. So it is concerned with changing and improving the heredity of the crops. Why we have to go? We'll see the objective in short. I will explain why we have to go for the breeding. So what are the objectives of the breeding? First one is the increasing the yield, high yield and earliness, increase the productivity. Why we have to? So this is the objectives of the breeding. First one is yield. It is concerned with the yield. Development of varieties and hybrids with the desirable characters. You know, in past there are different local varieties where there. Now there is a demand for hybrids. If you go in the market, 95% are the hybrids. Now you will not find the local varieties. So second objective, development of the varieties and hybrids with the desired characters. Desired characters means what type of characters you require, which is just preferred by the consumers. So that is where we say I will quote the example of tomato. It may be red lycopene contain or it may be beta carotene contain high, depending upon. So what are the desirable characters are there? Length, shape, size, color, these are the desirable characters. Then good quality. Quality is very important. Quality in respect of TSS is there. So acidity is there. Sugar acid blend is also there. If you consider the tomato crop, sugar acid blend is very, very important. Quality of the color of the fruit is very, very important. So color, then acceptance to the growers and consumers. So we have to develop we have to go for the breeding to develop the varieties and hybrids. Those are having very good acceptance by growers and consumers. Growers means so that growers can get the more benefit from the growing that particular variety or hybrid. Consumers, so that consumer can accept this in the market, preferably for eating it, for preparing it, means acceptance to the growers and consumers. Then once you see quantitative characters, breeding for quantitative character, increasing the yield. Second one is qualitative character for the quality purpose. Third one is then market acceptability, how the people are accepting, growers are accepting in the market. Fourth one is breeding for biotic stress. You know, this crop is infected by the real number of biotic factors, pests, insects, diseases, so many diseases, viral diseases. You know, now you come across the very serious viral diseases are coming on the most of the vegetable crops. So breeding for the biotic stress, resistant to different diseases, viral diseases, fungal diseases, bacterial diseases, then nematodes, all these breeding for biotic stress. Now the another sector is breeding for abiotic stress. You know, this is very, very challenging area. No doubt. If we don't have any resistant hybrid or varieties, we can go for the control measures are available for that. We can use the fungicide, pesticides here. But in case of abiotic stress, how we can go for this one? We have to develop the varieties we can tolerant to drought, tolerant to sunlight, high temperature, low temperature. Just I will quote the example of this year. In this year, there is a heavy rainfall. Intensity of rainfall is also high. Total rainfall, if I consider the Rauri situation, the rainfall average is 550 mm. It is more than 850 mm. So due to that, the crop get lost. So I will quote the example of onion. On large scale, this onion crop is lost due to the mist. You have to develop the varieties. Breeding for abiotic stress. Develop the varieties which can tolerate for. Say I'll quote the example of hot setting tomato, hot setting chili. We have to develop the varieties and variety which can tolerate and can withstand for high temperature, which can withstand for the drought, less moisture, which can also withstand for the flooding, high moisture. There are so many factors in abiotic stress. So the breeding for the abiotic stresses, then breeding for the suitability for the processing. We have to develop the variety as a hybrid which are suitable for the processing. Let's say it's a tomato. So most of the tomato different products are prepared. Paste is prepared, puree is prepared, ketchup is prepared. We have to develop the varieties or hybrid for the processing purpose. The purpose is processing. Then breeding for high nutritive value. You know, nutraceutical value. Along with your vitamins and mineral medicinal value. Some crop is also having medicinal value. You know, for the we quote the example of cruciferous crop. You can just see this one. So different crops, they are also having medicinal value. Tomato, beta carotene rich varieties also there. Beta carotene rich vitamin, all these things. Breeding for post-harvest requirement. So that you can see the storage life. Say, go to the example of onion. You have to develop the variety which can store for the long period. So that is post-harvest requirement. Maybe for basic purpose, we have to go for the breeding for specific purpose. Just you can say for long distance market, one of the specific purpose. We have to develop the varieties in case of tomato, which can withstand for long distance market. Then off-season vegetable. You know, in season we'll get the good price. But we grow this variety in off season. Just I will put the example of okra. Okra can be grown in Kharif and no doubt we'll find most of the vegetable throughout the year. But there are also certain vegetables because vegetables are the seasonal one. They require the certain climatic conditions, low temperature, high temperature, then long day plant, short day plant. So off season is very, very important because in off season we'll get the good market, high price, so that it is beneficial to the farmers. So off season, okra, no doubt in Kharif and Rabi, summer the crop, okra crop is good. But the okra variety which can withstand in the Rabi season, because if you see in the Rabi season, from October to January, you get the good price for okra. Means this is for basic purpose. We have to develop the varieties. Or say for the folios and shade now, you know it is vertical farming. You have to develop the variety in case of tomato, indeterminate type varieties also there. For malls and hotels, different star hotels, they require color capsicum, cherry tomato. Means we have to develop the varieties for wider adaptability. This will be widely adapted. So these are a few objectives why we have to go for the breeding in vegetable crops, because breeding is very, very important, which is achieved by two ways. One is the selection, another one is hybridization. So then self-pollinated crops. So what is meant by self-pollinated crops? Self-pollinated means pollination within the same flower. They will call it as a pollination, that is self-pollinated, transfer of pollen from anther to the stigma of the same flower. That is called as a self-pollinated crop. Just you have seen so many examples of the self-pollinated crops. Those are self-pollinated crops, or you have studied already. That is peas, bean, tomato. Sometimes in self-pollinated is also often cross-pollinated like a brinjal. Then what are the different breeding methods? So there are two components again here in breeding methods: general methods, special methods. So in general methods, plant deduction, pure line selection, mass selection, pedigree method, bulk method, single seed descent method, backcross method, heterosis breeding. In special methods, we can say it is the advanced methods in breeding: mutation breeding, polyploidy breeding, transgenic breeding, and molecular breeding. These are all both in your self-pollinated crops and cross-pollinated crops. These are the special methods. Then we'll see that by one. First one is introduction. Means introduction in new area, primary introduction and secondary adaptation. When the introduced variety is well adapted to the new area environment, it is released for the commercial cultivation without any alteration in its genotype. We can say it is a introduction. Introduction means in past, we have seen so many varieties introduced from foreign countries, maybe from, say, you can say the examples of different varieties in vegetable crops. So many varieties, they come from the different countries to our countries, that we call it as an introduction. We are just introduced without any alteration in the genotypes. As it is just grown in the particular area. Means it is adapted to the new environment, released for the commercial cultivation without any alternation in its genotype. Primary introduction. Secondary introduction. Selected variety may be subject to the selection or used in a hybridization program to develop suitable. You already seen so many. Already I told you there are two methods in case of this breeding. One is the selection, another one is hybridization. Once you got this material, you go for the selection. In even good plant, you can see it is different from that original material. Some few characters, they are improvement in the few characters. As it is, you can see the selection. Otherwise, it is used as in the hybridization program as a male or as a female on the basis of the desired character. We call it as a hybridization. Means there is some improvement, maybe by selection, maybe by hybridization. We call it as a secondary introduction. The procedure for introduction: once you procure the germplasm, you have to go for the quarantine, then evaluation, cataloging, multiplication, and distribution. These are the stages. So procurement of germplasm. We can procure any from any class. So there is NBPJR at New Delhi. Then germplasm for introduction can be obtained other countries also as a gift from individual, institution, exchange of germplasm by purchase or through exploration. There are restrictions on the export and introduction of plant material by post or introduction of germplasm from specific countries also there. There is one procedure, quarantine. If you bring it from the foreign countries, so you have to keep it, that is called as in isolation to prevent the spread of diseases etc. present in them to other material. So in case of plant introduction, all the propagules are thoroughly inspected for contamination by weed disease. Once you bring any material from other countries, you have to go for the quarantine process. So that that particular seedlings or seed should not carry any disease or any pest. Otherwise, if it come in our country, it may be spread of disease or insect. You have to go for the quarantine. The process is called as a quarantine. You have to keep this material in isolation. You have to see, examine whether there is a disease or contamination is there. We call it as a quarantine. This is the procedure. I will not go in detail. Then cataloging, evaluation, multiplication, just whatever the material you acquired, you have to go for cataloging it. You have to go for the evaluation of that material. Each germplasm evaluation consists assignment of germplasm assesses for their various features, traits of some known or potential use in breeding program. Each germplasm accession is given as an accession number. In India, the number is prefixed with the A, either IC or we can say EC. For indigenous collection, it may be within the country, we call it as IC, indigenous collection. If it is from abroad, other countries, exotic collection, we can say that is indigenous wild, IW. All information about species, variety, name of assistant, place of origin, adaptation are also recorded. You have to go for the germplasm register. Coding and decoding. What is the old name that is called as a coding? Then just I will say, if I bring any variety of the tomato from other place, I will do the code. Then I have to go, that is decoding, RHR tomato, this and this with the all specification in that germplasm register. That is, you can say it is a, that is a, cataloging is very, very important. We have to go for. The next method is a pure line selection. Very, very important. So what is the pure line? So the progeny of single homozygous cell-pollinated plant. That is called as a pure line. Mostly it is very, very important breeding methods in cell-pollinated crops. It is a progeny of single homozygous cell-pollinated plant. In a pure line selection, large number of plants are selected from cell-pollinated crops and harvested individually, and individual plant progenies from they are evaluated and the base progeny is released as a pure line variety. Just I told you. Anything, any material you have bought in introduction, you have to grow that one. Number of plants are there. You have to take the data of individual plant. If any single plant is different and having specific character with the desirable character, that you can go for the best progeny is released as a pure line variety, also known as individual plant selection. A pure line variety is a variety obtained from single homozygous plant of cell-pollinated crops and is maintained by self-pollination. So this genetic basis of pure line was explained subsequently by John and Sons in 1903. Then what is the procedure? Just I will explain in short. 2,200 to 3,000 plants are selected for superior. You can grow it randomly. So you have to select the individual plant. Individual plant produces in second year. Just in first year, you grow the population. Whatever it may be, 200, 500, 1,000. Then you select on the basis of phenotype. Phenotype means that is a morphological character. What is the intention or objectives of that breeder? Why for that breeder is going for developing the variety. On the basis of desirable character, you have to select few plants, individual plants in first generation. In second generation, you have to grow the individual plant. Individual plant produce are grown, and whatever the undesirable produces are that we have to reject in second. Means you have to take the data individually. Then again in third year, selected produce are planted in preliminary plant. Whatever you got good, you can grow in third year, individual plants. Again in third year, inferior progenies are rejected. In 4th to 6th year, you have to go for the vertical. What you got up to third year, good 4 or 5 good lines, that you go again replicated trials and suitable checks are conducted several locations. We call it the vertical. At least you have to go for the 4 locations and 2 year data. Total 8 trials are required for the releasing of the variety. First year we go for release, second year we go for the release. So up to 4th to 6th year, 2 year, you have to take the data. Multi-location means just it is the variety from Rauri. You have to grow at Thode, Pune, Kolhapur, or say Nandurbar. Different locations. Means if you have to see, though the genotype is same, if it has any effect of the that particular environment, because we are releasing the variety for this particular jurisdiction of the 10th district or Western Maharashtra, throughout the Maharashtra, you have to select the locations, you have to conduct the multi-location plan. Here you have to take the data, yield data, and you have to see that disease resistance, incidence of pest, and all these data should be. And along with check any good variety, you have to grow that one. And then in 7th year, you have to go for the releasing variety and then seed. So it's very simple method, pure line selection. Progeny of single homozygous plant is called as a, this is the very simple process and this is widely adopted in the selection procedure, that is in improvement of the vegetable crops. Application of pure line selection, improvement of local variety. Whatever the varieties you see now, improved varieties, mostly those are all selection, pure line selection. If you see in your course in vegetable only, you may study so many varieties are there by pure line selection, which are developed from the local varieties by adapting this portion. Improvement of local varieties, selection in introduced varieties, improvement in old pure line varieties, selection of new characteristics, pure line. On the basis of desirable characters, you can develop the new variety. These are the advantages. Best pure line present in the population, we can give the variety. Extremely uniform because it is a pure line selection from individual plant, it is uniform and preferred by the farmers, consumers to less uniform variety developed through the mass selection. Due to its extreme uniformity, is one of the requirements for registration of the variety because you get the uniform material by the pure line. What are the disadvantages? The variety developed through pure line selection generally do not have wide adaptation or stability in production. Pure line selection require more time, space, and it is expensive trials than the mass selection. They definitely, but pure line is the best. The improvement is depend on genetic variation. Yes, because improvement is depend on variability. If there is a genetic variation among that population, then and then we will get the different variety. The breeder has to devote more time, yes, for pure line selection than because it requires 7 to 8 years. Mass selection, next method. Large number of plants having similar phenotypes are selected and their seed are mixed together to constitute the new variety. The variety developed through mass selection or pure line selection may be done in such variety. See this one. In pure line selection, single homozygous plant. Here in mass selection, the large number of plants, those are morphologically similar. Means their phenotypes on the basis of similar phenotypes. Phenotypes mean morphological character. They are selected. Their seed are mixed together and this constitutes a new variety that is called as a mass selection. What is the procedure of mass selection? Again, 1, 2, 3 or 4 we have to go for this procedure. From variable population, you may select 500 to up to 2000 plants with similar but desirable traits are selected and seeds from the selected plants are calculated. See this. In the first year, on the morphological characters, similar but desirable traits, they are selected, they are grown, their seed is mixed. Second year, the composite seeds are planted in preliminary yield along with standard check. Phenotype of selected population is critically. Again, you have to grow this particularly, whatever the material is there, along with the standard check. Standard check means any good variety, commercial variety, or in hybrids, it is commercial hybrid. But here, in case of mass selection, any good variety, it is taken as a standard check for the company with that, it is superior to that particular variety or not because we are going for the improvement by the mass selection. Phenotype of the selected population is critically evaluated. You have to evaluate that one. Then, 3rd to 5th year promotion selection are evaluated in quantitative yield trials in several locations that you have to go again multi-location trial along with the superior check. You have to take, once you have taken the data of the station where you are developing that variety, you have to take the data for 3 years, multi-location data. Maybe from Dhule, Pune, Kolhapur, and you have to see that whatever the material you are going to release is comparable, you can develop a very good variety rather than whatever you are having in your own varieties and it is superior to the whatever the check you have taken. And then in 6th year, seed multiplication. What is the difference between pure line and mass? Pure line, progeny of a single plant. Here, similar characters, different plants, they are having desirable characters, morphology, they are mixed together, it is grown and then it is developed as a commercial. Advantages. Mixture of closely related phyllodes is more stable in performance of different environment than a single because it is a variable. You can see it is a mixture of closely related pure lines. Often the extension and prolonged yield trials are not necessary. It reduces times and cost. Mass selection retain genetic. Yes, it is a mass selection. You on the basis of you can say similar characters you are mixing, have genetic variety in new variety. It is a less demanding method, but see this one. Though this having some good advantages, it is the less demanding. Mostly pure line selection method is used in a cell formative. Then disadvantages. Not that infer as a pure line. Yes, may some variability is there on the basis of permanent, hence less like the pure lines. The improvement through mass selection is generally less than that could be achieved to the pure line. You cannot get so much improvement through this mass selection. It is not known that if the phenotype strict superiority of the selected plant is due to environment availability. Yes, we can't predict it. Whatever the superiority is there in this particular mass selection, it may be due to the genotype or maybe the phenotype environment. It may be due to the genetical constitution or phenotype. You can say environment. Due to the polarity of pure lines, mass selection is not commonly used in place. I already told you. Variety development and mass selection are difficult to identify than the pure lines due to some variation. Is that? Then pedigree steps is the best methodology. It is also important. Yeah, pedigree method. So what is the pedigree method? In pedigree method, a detailed record of the relationship between the selected plants and their progenies are maintained. Such a record is known as pedigree record. Why this method is called as a pedigree? Because we are maintaining the record of the different generation from F2 to F2, 7, 8 up to the stability in that material. So hence it is called as a pedigree. Pedigree means the record is maintained here for all generation, individual plant record. It is recorded. That we call it as a pedigree method of this particular breeding of cell. What is the procedure? So here is like that. See this one. You have to select two parents having desirable characters. Say I have to develop the, and by that way, we are developing inbred lines by pedigree method. Inbred lines. We can see it as the transgressive segregation. You know, we are going to develop the different segregants. Combination of the characters. You know, here pedigree. You have selected female having very good yield character, earliness, all these things. You have selected second parent. It is also having some desirable characters. Your crossing is selected parents are planted in a block. First one is on the basis of their characters and the breeding objectives, you have to select the parent. In breeding, it's the responsibility of the breeder to select the parent. So, on the basis of what is the objective of the breeder to develop the variety, on that basis, or on that basis, you have to develop this lines. So, you have to select the parent. So, in my mind, I have to develop the lines different, having very good yield, very good quality characters, resistant to different diseases and insect, all this. So, what are the important objectives? On that basis, you select the parent, female and male, P1 and P2. P1 is always female, P2 is always male. So, selected plants are planted in the block and crosses are. You have to go for the pollination. You have to go for the pollination. We'll get the F1. F1 you get the F1. It is a homozygous. F1 is there is no variation in F1. So again in second year, you select 10 to 30 seeds and are planted in F1. So if you are in F1, then in again F1 you select so many plants, 2000 to 1000 plants, depending upon 100 to 110. If you select more number of plants, because you will get the segregation in F2 and F3, later on you will not get the segregation. You have to go for the transgressive segregation. What is meant by transgressive segregation here? These are the progenies which are superior in 1 to 2 character than the parent is called as a segregants. You will get the segregants in F2. Different variation you will get in different individuals. So you have to grow more number of plants. So in this particularly 2000 to 1000 plants are selected in third year in F2 generation. Then 100 to 500 superior plants are selected from this particular generation. You go for F3. Here individual plant progenies are best planted and superior plants are selected. What you have to follow from F3 generation? You select in F2 generation single plants. You have to grow single plants in F3 generation. Whatever the superior plants, you go on selection. And keep the seed of single plant individually. On the basis of phenotypic character, as per the mind in the breeder, on the basis of desirable character, you select the plants having the desirable character. Superior plants are selected from the superior progeny. F3 in 4th year. Then same again in F4, you go for selection of the superior plants. In F4 again, F5 individual plant progeny are planted in multi-row plot, superior plants are selected from superior progeny. In 6th year, we'll get the individual selections. And up to 6th year, we'll get the plants which are stable. So there should be any variation. Up to Fourth, fifth year will get the variation in that particular. So in sixth year, we will get the desirable plants which are stable in character, which don't have any variation in the characters. So it requires up to 6 years. You are maintaining the record from F2, F3, F4 on the basis of desirable character. Every time you have to take the data of individual plant. How much plants? Depending on different crops, 100, 200, 300, 500. So that you will get the different lines. We may say the inbred lines or segregants, transgression segregants. You will get all this, and from that you select the good lines and you go for the variety development of the variety in 7th year. That is preliminary yield trial. What you got? 10 to 12 good lines having very good desirable characters as per the minds of the breeder. No, I got such and such line which is as per my objectives. Then you go for the in 7th year, that is in F6 just preliminary yield trial, in F7 preliminary yield trials, different testing, and then multi-location trial, and then release of variety. So combination of the characters of the two varieties, P1 and P2, and you are maintaining the record. You are getting the characters which are superior than the parents because it is a transgression segregants that you will get in the at least in 7th year. And if you got any good lines, you can go for the release of variety, and you are maintaining the record. Hence, it is called as a pedigree method. I will take directly. Then coordination trial and all this. So maximum opportunity to the breeders to use the skills, judgments for selection. Yes, it is very important. You will get the good judgment for selection. The breeders, desirable plants, particularly early segregate, earliness, yield, their size, shape, color, depending on the type of the crop and characters. It is well suitable improvement of easily identified and simply inherited characters. It takes less time than the bulk method. That are we often able to obtain information about the inheritors because you are maintaining the pedigree record. Maintenance disadvantage. Maintenance of pedigree record takes up valuable time. Selection among and within large number of projects vary generation leverage. Success is likely depend on the skill of the breeder. Yes. Breeder skill is very good. How with this eye he is on the basis of character is selecting the material is very good. There is no opportunity for natural selection. Bulk methods very important method again is the first Nelson and Lilly in 1908 extra. So in bulk method, see this one. In pedigree and bulk, F2 and subsequent generation, it harvested in a mass or bulk and raised to next generation. At the end of bulking period, individual plants are selected and in a similar manner as in the pedigree. So here, at the end of the bulking period, you have to select here. See this procedure. Again, you are going selected plants are hybridized. F1 planted seeds are harvested in bulk. Plant are commercial seed rate harvested in bulk. Same in F3, F4, F5, F6. Then F7 space planted superior. You have to go for mixing bulk method in every generation. In case of pedigree method, you are growing individual plant. Here, every generation, whatever the individual plants you are getting, you are bulking and growing for again next generation. Up to F7. In F7, you are selecting the superior plants and harvesting separately and going for the evaluation, multi-location and all these things. Simple convent experience method, inexpensive method and convent. Eliminate undesirable ties and passive isolation. Little work and attention is needed because there is no so much exercise. Because in pedigree, individual plant selection record is record. Here, up to 7 generation, you are going for mixing. It takes much time to develop new variety, provides very little opportunity to the breeder. A large number of progenies are handled. Information of inheritance. Here, no any information of inheritance. Transfer of character from progeny is not maintained here. Single seed descent method, very, very important. It is the modification of bulk method. This method is particularly suited to develop population of recombinant inbred lines, just I told you, inbred lines. So, formed by crossing of two inbred strains, followed by repeated selfing and dibbling mating to create new inbred lines, whose genome is mosaic of the parental genome. So, procedure of selected parents hybridization. F1 place planted and seed harvested in bulk. So you have to densely planted from each plant, random seed is selected. What is the area of 2? So you select the seed from a single seed from a plant. Seed is selected and again bulk. F3, F2 densely plant from each point, one random. You have to select a single seed from, that is single seed this method. As in F3. In F6, whatever the plants you have selected, space planted 100 or 500 plants with desirable characteristics are space planted. And in F7, individual plant progeny are grown, and whatever the weak and undesirable progenies are there, single seed descent method. Every generation, you take the seed from single plant, single seed, grow the progeny. So in this way, that is single seed descent method. Backcross method. This is very, very important method. Maybe in self-pollinated and cross-pollinated. You have seen here, already in my objectives I mentioned, breeding for qualitative characters, quantitative characters. But when we have to go for the disease resistance, pest resistance, this backcross method is very, very important. A cross between a hybrid F1 and one of his parent is known as backcross method. The hybrid and the progeny, the subsequent genus are repeatedly backcrossed to the one of the parents of the F1. Which character you have to introduce? Means here, which character disease resistance we are using male as a resistant, that is donor parent here. Repeatedly we have to go for addition of genes, you can say here. Means just I will show the procedure. So recipient parent, which is repeatedly used in the backcross program, is generally known as recurrent parent. The donor parent on the other hand is called as a non-recurrent parent because it is only used once in the breeding program for producing F1. So you can say recurrent and non-recurrent parent. So what is backcross? Crossing between hybrid and is one of the segregated, we can say parent is known as backcross. And this is the best method for the development of the disease resistance lines of hybrids. Procedure of backcross. See this one. Non-recurrent parent female, recurrent male. So now non-recurrent parent you use once only for crossing. It is a female. Recurrent parent male. So you can see F1 is crossed with the parent, say this one, BC1, will get the 50% A resistance. In second, BC2, 75% resistance. In BC3, 87.5% resistance. BC4, 93.7%. BC5, we can say 96.87. And in BC6, 90. So that will get the resistant mutant. You are adding the gene from recurrent parent. So here, the responsibility of breeder is you have to select the resistant parent is very, very important. Because so that you will get the good material that is in BC6, which is having very good. So say that your female parent is this variety is very good in your yield and all these things. This variety is susceptible to the different disease. What is your important disease? I'll go to the example of tomato leaf virus disease is there. So I have to take the parent, so recurrent parent which is resistant to leaf curl. So that I will go for addition of genes. Let's see this one. So that in BC6 I will get the good material which is highly and resistant to the disease. That is the procedure of backcross method for transfer of genes. See this one. This is one of the example of rust resistant variety. How it is will get the genes addition of 50% gene, 75% gene, 87.5% gene, 93%. This is mostly useful for the resistant breeding program, this backcross method. No doubt now we are coming across, we don't have time, otherwise transgenic is coming on. So transgenic, we are transferring the genes, but this is the traditional method in past. Most of the breeder has used this method. This method has been used by most of the breeders. This was the only one method, backcross method for going for the, we can say for the resistant breeding program. You can go for the adding of the genes, which is responsible for the resistance. So resistant in the particular very good variety. See this one, at last we'll get the 96.87% genes from A. The new variety is nearly identical with that of recurrent parent, except the gene transfer. Thus, the outcome of backcross is non-forward B forward. That you can say B forward. Not necessary to test the variety in extensive yield rates. The backcross process is not dependent on the environment. Much smaller population is needed in this method than the pedigree. Defects of well-adapted variety can be removed without affecting its performance. Yes, whatever the defect are there, say we have used the female A, which is susceptible. So we can go for the addition of transfer of the gene that is resistant. The defect what will it can be removed. So disadvantage generally cannot superior than the recurrent parent except for the character which are transferred. There may be undesirable genes closely linked with this being transferred, may be transferred to the new variety, producing linkage drag. By the time backcross program improves, the recurrent parent may be replaced by other improved superior variety. So these are the disadvantages. Then heterosis breeding, very, very important method in this particularly breeding methods, maybe self-pollinated crops or cross-pollinated crops. So nowadays, you know, this there is a scope for the heterosis, you can say hybrids. So term heterosis is first used by the Shell in 1914. Heterosis may sometime defined as the superiority of F1 over both of its parents in terms and yield of other characters is called as the heterosis. See this one. Heterosis, development of that by hybridization program, means Whenever you get the hybrid, it is superior than its parent. So it is called as heterosis. Superiority of F1 hybrid for both its parents in terms of yield. Maybe in the terms of yield, or maybe it means superiority, maybe in the terms of quantitative characters or qualitative characters, that we call it as F1 hybrid. Maybe this is resistance. We call it as a hybrid having good quality, good yield, earliness along with the certain, this is resistance. So you can say heterosis. Mainly cross-pollinated crops show heterosis, but in sun-pollinated crops also show heterosis, but it is a magnitude is generally because in sun-pollinated crop there is a less scope for heterosis, and in cross-pollinated crops the heterosis is not more than 20%. But in case of cross-pollinated crops, you may get the higher percentage of heterosis. This method of breeding is used for the development of many commercial hybrids. So many hybrids are there which has been developed by this particular mention. So as you can see in this particularly MPK has developed so many hybrids, because due to the less time I have not given the example of pure line, pedigree, or so many vegetable crops, so many vegetables in crop varieties in self-pollinated and crop pollinated crop are developed by different breeding methods. But in case of hybrids, I got some developed varieties in case of tomato. This the tomato improvement scheme has developed the hybrid that is Phule Raja, which is indeterminate type. In case of brinjal, we have developed Krishna, Phule Arjun. So many hybrids have been developed by this particular. So in case of sponge also Phule Komal has been developed. So in vegetable crops, our MPQ has developed so many hybrids that I could in our country also. Indian Institute of Horticulture Research IHR Bangalore also developed so many hybrids. PUSA also IR also developed so many centers are there. Those have developed so many hybrids and varieties using different varieties. So this is really important. So heterosis again, this method of breeding is used for the development of many commercial hybrids. Just I told you, if you go in the market, for your knowledge, 95% hybrids are there and 5% are the local variety. In few cases, onion, not so popular hybrids are there. All varieties are popular. Some options, but now hybrids are also coming in onion. See what is the difference between hybrids and varieties, just I would like to quote. Because the cost of production of variety is less rather than the hybrids. The cost of production of hybrid is more because you have to go for the every time crossing, and the cost of production of the seed is very high. But there is a scope because the hybrids are very good in yield. Registering to go in the market. Hybrids are very good desirable characters, not for yield, but for qualitative characters, for the abiotic stress and biotic stress. In heterosis, some terms are very important. Heterosis is more generally heterosis, which is estimated between related to descent density. In case of the self-pollinated crops, there may be the chances of inbreeding depressions. That is, inbreeding depressions may be defined as reduction or loss of vigor. Because we go for the heterosis, there is definitely you get the, because hybrids are with vigor and very good. It is vigor. Vigor is very important here. We get the very vigorous hybrids. And here we go for inbreeding depression may be occurred in the particular self-pollinated crop. So what is the luxuriance? Is the increased vigor and the size of interspecific hybrids. Because here we are going for the interspecific hybridization, means between the different species, two species of the same crop. Then breeding methods in cross-pollinated crops. So population improvement with progeny testing, mass selection, without progeny testing, progeny selection and recurrent selection, hybrid varieties, synthetic varieties, so many varieties and different things are there in this particularly. So population improvement, mass selection or its modification are used to increase the frequency of desirable animals, thereby improving the characters of the population. So this population improvement may group into following general classes. Without progeny testing, mass selection. With progeny testing, your Toro method and recurrent selection. Mass selection, same like again in this particular self-pollinated. Number of plants are selected on the basis of their plant. And open pollinated seeds from them is bulked together to raise the next generation. Here, on the basis of phenotypic characters, which are the desirable characters, similar characters, they are selected and open pollinated seeds from them is bulked together to raise the next one. They are allowed to pollinate because it is a cross pollination. Open pollination is there. So, and you will get the next generation. It bulk together in the next generation. Progeny selection. Here to row method. This method is developed by Hopkins in 1908. It's the simplest form of progeny selection. This method is extensively used in maize. It should be in different, just it is the example of the vegetable crops also it has been used. To overcome this drawback of modified year-to-row method has been developed. Year-to-row method. See this one. Plants are selected on the basis of genotypes. First year, open pollinated seeds from each plants are harvested. The plants are selected, they are allowed to pollinate, and again, the open pollinated seeds from each plant are selected. In next generation, small progeny rows grown from the selected plants, superior progeny is identified and selected. In second generation, plants allowed to open pollinate, seed is harvested separately. Again, same in second year. Then in maybe repeated one or more times and yield loss. Means here progenies are selected, superior progenies are selected, and again we are going for the next generation. Year-to-row method. Plants are selected on the basis of phenotype and open pollinated seeds are from each plants are harvested safely. It is again grown in next year for us. Again superior plants are identified. Again it is harvested. Plants are allowed to open pollinated seed is harvested safely. Here you are growing the selected plants. They are allowing to pollinate open pollination, and you are selecting individual plants. Same we are repeating this one. This is called as year-to-row method. Here there is a chance for variability by the open pollination. Though you are selecting progenies, but they are allowed to open pollinate. No any barrier for the restriction in the pollination. Advantages. Selection is based on the progeny test, not on the phenotype. Inbreeding may be avoided in case if care is taken. Selection scheme is very simple. Selection is based on material only, which reduces efficiency. Yes, because whatever the material is there, on that basis selection is there. And superior progenies you are selecting. Many of the schemes are complicated in one considerable mark. It is selection takes place twice as much time is in mass selection. Modification of year-to-row method. Some modifications are there. What is the modification here? Plants are selected on the basis of phenotype. Open pollinated select seeds plant, each plant are harvested. Here, small progeny rows grown from the selected plants. Superior progeny is identified, remaining seeds from plants selected in first year, producing superior progeny to the next generation. What we are making here, modification. Whatever the remaining seeds are there, selected in first year, they are bulked together, producing superior progeny to the next generation. And this plant producing superior progeny, the bulk seed in second year planted, plants are allowed to open pollinate, and plants with superior phenotype selected and seeds are allowed. Means we are giving the chance to create the variability. Whatever the bulk seed is there, you will get the some variability, and you are going to again pollinate them. Again in second year, this is called a modification. In year-to-row method, you are individually selecting. Here, whatever the remaining seed is there from plant selected in first year, we are mixing together, bulking together, and in next year, again we are going for the sowing. So that we will get the population. Again, we are selecting the individual plant. This is the only modification. Recurrent selection. In cross-pollinated population, schemes of selection followed by intermittent the selected plants. So this is the recurrent selection. I will not waste so much time here. Selection, I have to show you the mutation program. This is the recurrent selection, reciprocal recurrent selection. Then hybrid varieties. What are the hybrid varieties? You can see the hybrids. So first generation from cross between two pure lines inbreeds open pollinated variety or other propagation, which is genetically different, that is called as hybrid, or in market it is called as hybrid. We can say this is the hybrid. Two terms are there, hybrid or hybrid variety. In general market, we people are saying, some people are saying hybrid variety. What is it? It is nothing but development of the hybrid by using two pure lines or two inbreeds or open pollinated variety, clone or other population, which is genetically dissimilar. Genetically dissimilar means having variability. Say I can say one is very good in yield character but susceptible to the disease. Another is not having so much superior fruit character but it is resistant to disease, pest, or having very good qualitative characters. Means those are genetically dissimilar from the same crop. And we are making the combination of the character, combination of the gene, which are genetically dissimilar. When two inbreed lines are crossed, it is called as a single cross hybrid or single cross. When two single crosses are crossed, it is called as a double cross. Means here A plant is crossed, A variety is crossed with B variety, single cross. When AB is crossed and CD is crossed, that two crosses again made, that is called as a double cross. Three-way cross is a cross with single cross and a one inbreed or one variety. Means you have crossed A variety into B variety cross, and again you have crossed with C line or inbreed line or any local variety. It is called as a three-way cross. Specifically in market, we are going for the single cross development of it. A is crossed with the B. For research purpose, we are going for double cross, three-way cross for development of segregating population having the variation in the characters. When an inbreed is crossed with an open pollinated variety, it is known as inbreed variety cross or top cross. See this one. What is meant by top cross? We'll come across when we come here for MSc or PhD. We'll come across with these terms. What is top cross or inbreed variety cross? When in inbreed is crossed with the open pollinated variety, it is called as an inbreed variety cross or top cross. Polycross denote the progeny of a line produced through the random pollination by number of cells. Polycross means you are crossing with n number of lines. Polycross, poly means many. Random population, random pollination by number of lines. It is called as a polycross for development of variability. When a cross is made to assess combining ability, it is called known as a test cross. When a cross is made for assessing combining ability means which is the best parent. If you go for the heterosis breeding program, you will come across heterosis and combining ability. General combining ability and specific combining ability. Two things are there. Then when two open pollinated varieties are mated, it is also known as a varietal or population cross. Because you are making crosses of two open pollinated varieties, which are popular varieties, well-known varieties, that is called as varietal or population cross. Procedure of hybridization. First one is the choice of parent. So this is very, very important nowadays. Hybridization program is fun. We can say development of hybrids or hybrid varieties. But as per the objectives of the breeder, you have to make the choice of the parents. On that basis, that parents should have the desirable character. So what is in the mind of the breeder? What is required in the market? Which are scoping the consumers? Which are the scope of the growers? What type of hybrid is needed by the farmers? And which are the say acceptance by the consumers? On that basis, which parents you have to select for the hybridization for development of hybrid or hybrid variety? That is the choice of parent. You have to select very good parent. You select the parent, you go for the evaluation of parent. You have to take the character of the parent. No, whatever the characters given in the record, it is right or not, you have to evaluate the parent. If you evaluate the parents, you go the front. So there are different methods are there. Then you go for the emasculation. In that particular flower, if it is say mono, you can say this a flower, you have to go for the emasculation. Removal of the anther, we call it as emasculation. Then pollination. In this particular cross-pollinated crops, both the, you can say the anther for male and females are different. You take this particularly male, the cross-pollinated crops, you can see this one. You go for the emasculation within the flower, so you can remove this particularly. All these anthers go for pollination, transfer of pollen, go for tagging, bagging. You'll get the seed. It will be the yacht one. This is the procedure. Choice of the parent, evaluation parent, growing of the parent. Go for the emasculation, remove anthers, go for pollination, you go for the transfer of pollen from other variety. So if you remove the anther of that particular female, it will be that flower, it will remain as a female. You transfer the pollen from the male, will have the pollination. Then you go for the tagging. Sometimes you have to close that particularly flower, maybe by cotton bug, maybe by bagging. So many methods are there, so that there should not be continuation of other pollens. Because this is a cross pollinated. So just to cover with the polybag, polythene bag, or maybe cotton, or maybe just tie it, so that there should not be contamination of the pollen by other lines or other animals. Tagging, tag it. But which type of cross is there? On which day you have made the crossing? Go for the bagging, bag it, and then you get the fruits, you will get the seed. Go for the harvesting and storage of F1 seed. Then synthetic varieties. So you got a hybridization program. It is very, very important. Nowadays there is a demand from these hybrid varieties. We have to follow this method, that is development of hybrid by this method, hybridization program. By that way, you can cross two lines, maybe open pollinated varieties, inbred lines, pure lines with the desirable characters, with the specific characters which in the mind of the breeder, which have the scope in the market, and there is a preference from the consumer. Synthetic varieties, it's produced by causing all the combination number of lines that combine well with each other, maintained by open pollination in isolation. Composite variety, produced by mixing the seeds of several phenotypically outstanding lines and encouraging open pollination to produce cause in all the combinations among them. That is called as a composite variety and synthetic. Just I will give this is the last. What is the synthetic variety? Sometimes this question is asked, but students are not able to give the difference between synthetic and composite. The synthetic variety is produced by crossing all the combination of number of lines that combine well with each other and maintained by open pollination. They are maintained by all of the open pollination. What is composite? Here it is produced by mixing the seeds of several phenotypically outstanding lines. Phenotypically outstanding lines, and then encouraging open pollination to produce the cross in all combination among. This is the difference between synthetic and composite variety. Combination variety is produced by mixing the seeds of several phenotypically outstanding lines. That is called as combination. It is produced by seeds of several phenotypically outstanding lines. Combination variety. Mixing procedure. This is the procedure. I will not waste my time. This is all about the presentation here for the self-pollinated crops and cross-pollinated crops. But there are very important 3 methods. 4 methods are there, special methods, but due to the time, I could not complete that one. Just I will give the what is these are the special methods here. That is, one is the polyploidy breeding. Polyploidy breeding is very, very important nowadays so that we can develop the polyploid. The best example of polyploidy is watermelon. Then you can go for the mutation breeding, change in the genetic makeup of the plant. You can see mutation breeding is also very, very important. Then transgenic. Nowadays, the transgenic breeding is very, very important because in past we are using the method for transfer of gene for any say specifically for the disease resistant by already I explained you backcross method. But here for backcross method also you require that the species or line of that crop. What is the difference between this? In transgenic you can transfer the gene from anyone. So many transgenics are there in the market. So in BT brinjal is there. Already you know we are concerned with the BT cotton is also there. BT okra is also there. Transgenic transfer of gene in tomato flavor saver is there. Nowadays the transgenic is coming. It's a more advanced method of breeding. But due to the less time I could not explain because I am also having that much presentation with you. But just these are the very for you student the basic methods of breeding of vegetable crops for the cross-pollinated crops and the self-pollinated crops. Okay, thank you.
Thank you, sir. Thank you.
1 minute, sir.
Yes.
I'm closing this. 1 minute. 1 minute, sir. So take this. Kabra sir, please. Kabra sir, please. Hello.
Thank you for very nice lecture. You. I'm very belong of the College of Agriculture. I am very thankful to Dr. Shivsagar sir. Thank you, sir. Thank you.
Okay, thank you. Because what happened? I have so much material, but due to the time, I could not manage this because this is for basic. Otherwise, advanced methods are very, very important. But these basic methods I have got because within 1 hour it's very difficult. In mutation breeding, transgenic breeding, polyploidy breeding, these are very basic and advanced methods. But for UG people, this okay. These basic things for which are those methods is very, very important. Okay, thank you. Thank you for giving me this opportunity to deliver the lecture. I thanks Dr. C.D. Devkar sir, Ashok Dean, College of Agriculture today, and my friend Dr. Kabra sir for giving me this opportunity to deliver the lecture to the students of College of Agriculture today. Thank you once again. Thank you.
Thank you.
मला याची युट्यूबवरची जर रेकॉर्डिंग असेल तर प्लीज पाठवा बरं का माझ्या व्हॉट्सअपला.
Yes, sir. Yes, sir.
याची रेकॉर्डिंग असेल ना आता तुमच्याकडे?
Yes, we record it on YouTube, sir.
Okay. Please send me that one also. Okay. Thank you. आता कुठलं लेक्चर आहे? याच्यातून मी हे करू का आता बंद करू का हॅलो
आता 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 आता
हॅलो.
Hello. Huh? Ito na po. Daw. Iyo, na po. Next one.
I got it.